All right, everyone, we are live. Welcome to Stemworld. My name is Diana Papasanel, and I'm the CEO and co-founder of STEM Enrichment Youth, the nonprofit that is hosting this organization. Off to Catherine.
Hi, I'm Catherine, and I'm the co-founder of STEM Enrichment Youth.
Hi, I'm Aryan. I am an executive member of STEM Enrichment Youth.
Hi, I'm Sophie. I'm also an executive member of STEM Enrichment Youth.
Awesome. Stemworld is a completely free two-day convention that captures the highlights and intersections of biology, chemistry, technology, mathematics, engineering, and physics. We will dive into incredible hotbeds of innovation, from nanotechnology and black holes colliding at the edge of the universe to quantum computers and the application of computer science in decoding the human genome. Stemworld's goal is to encourage people of all ages to explore diverse mathematical and scientific disciplines and to educate them on various STEM concepts and marvels. With Stemworld, you'll be exploring countless areas of science and technology for a maximum of 22 hours over the next 2 days, with the freedom and flexibility to attend any event that appeals to you. With its two main components, speaker events and competitions, Stemworld is a great way to promote learning and educating during the pandemic, when many summer and fall STEM programs have been canceled. We are separating most of our events by subject, so today we will delve into biology, chemistry, and mathematics, and focus on physics, computer science, and engineering tomorrow. For each subject, we have a panel of 5 STEM professionals, including many accomplished university professors, researchers, and PhD candidates. Dr. Sonia Suelar Ortiz and Dr. Jason Graham will be hosting our biochemistry and computer science demonstrations for us on Sunday. A special thank you to Schmall Science Workshops for helping us get these demos up and running. With the COVID-19 lab and safety protocols, it's been extremely difficult for most students to explore hands-on science in the pandemic. Our hope is that these demonstrations can help our attendees explore and advance science from their own homes. In addition to our professional panels and demonstrations, we are also hosting 16 undergraduate speakers from the top US universities to discuss their high school and college extracurriculars that encourage them to pursue and master their skills in STEM. They'll be helping us get an idea of what these top ranking colleges are really looking for. So if you're gearing up for college app season, be sure to check them out tomorrow from 3:30 to 6 p.m. PDT. William Lid, an MIT undergrad and IOI finalist, will be joining us all the way from Taiwan at 6:30 p.m. PDT today. He's the only undergraduate student going today. Stemworld also has 3 networking sessions where you'll connect and engage with STEM enthusiasts from over 15 countries. Participate in icebreakers, they'll be fun, I promise, and discuss your favorite parts of the convention. Kimberly Weifling, an amazing leadership consultant and physicist, will be joining us for these sessions to get the party started. We're also hosting 4 high school STEM competitions for physics, biology, chemistry, and mathematics. The times and descriptions for each competition can be found on our website at stemenrichmentyouth.com/STEMworld. If you want to compete, please join our Slack workspace at tinyurl.com/STEMworldSlack, where we will be sending out the links to compete. The top 9 competitors of each competition will receive prizes, which will be available to view in our Stemworld Slack workspace. Please bear in mind that our competitions were written by high schoolers, so while our writers are incredibly knowledgeable in their subjects, they're still only in high school. Now, an incredible thank you to all of our sponsors that made these competitions and all of these prizes possible, including Next Step and Carry the Vision. To introduce them more in depth, we are going to hand over the floor to Next Step, one of our platinum sponsors. Adozi is here today to join us and talk a little bit about Next Step.
How's it going, guys? So my name is Adozi Ezeanway. I'm a junior at the University of Notre Dame. I am the founder of Next Step. I'm actually in my dorm room right now because we are actually one of the few universities to actually come back to campus. So when I was in high school searching for which colleges I wanted to attend or even apply to, I had a very hard time, deciding which university would actually be a good fit for me. Everything I looked up was either statistic based or possibly from an untrustworthy source. You couldn't really, be sure that that person actually went to that school. So one thing I always wished I had was the ability to talk to someone like me that goes to a college I'm interested in and just chat about whatever I wanted, whether that be how the food is, college application advice, tips, etc. So ultimately, I created just that. Just so I don't end up blabbing on too much about what Next Step is and what we do, I had a short explainer video put together for you guys. If we could put that up. Let's see. Should I just go ahead and put that up?
Oh, yeah.
All right, for sure.
Yeah, I'm just gonna make you close so you can share your screen.
Yeah, of course. Let me get that for you guys. Here we go.
Yeah, let me know if it works.
Sharing screen feature is not working for me.
I have the video. I can put it.
All right, perfect. Thank you.
James is a high school student, and just like many other students, he is stressed and confused about which college is a good fit for him. He and his parents are tired of spending thousands of dollars traveling and touring schools just to leave with information he doesn't really care about. He tries to look up the schools online, but all he can find are rankings and reviews that could easily be faked or are from untrustworthy sources. Luckily, James found Next Step. Next Step is the only service that facilitates a 1-hour one-on-one live conversation with a student attending any college that you're interested in. Here at Next Step, we match you with college students that resemble your future self and recommend schools based on your personality, interests, major, and more. Want to know what the social scene is like? How the workload is for your intended major? When talking to your personal Next Step mentor, you get to ask the questions that matter to you. Why spend countless hours and thousands of dollars when you can get all the information you need in a much more convenient, affordable, and comfortable way? Visit our website and see why people choose us. Next Step. Find your perfect fit and sign up today at www.nextstep.co.
All right. Yeah, that pretty much sums it up. So yeah, basically once you go onto the site, you make an account and you fill out your Next Step profile. Once you fill out your Next Step profile, this is what we use to gauge and match your personality with other college students going to the college. Right now, all first meetings are free. And yeah, I want to thank STEMI for giving me this opportunity. Thank you guys.
Yeah, thank you so much, Adozi. We know that college app season is already here and many high schoolers are struggling to navigate the super dense and complex college application process that would incredibly benefit from Next Step services. So we will post all of the information that Adozi just reviewed, including the video and all of the links for Next Step on all of our social medias and our Slack workspace. They're also in the chat and in the closed captions if you want to see those. So be on the lookout. And last but definitely not least, we would like to give a huge thank you to our fiscal sponsor for this convention, Carry the Vision. We'll hand over the floor to CTV or Carry the Vision so that they can talk about what their organization does and how you can get involved. So please welcome Girish Shah, and as he discusses Carry the Vision.
Good morning, everyone. In this day and age, you use Zoom so much, you still forget to unmute yourself. I welcome you all to this workshop It's a wonderful workshop that I learned only a few weeks ago. And I hope that you all attendees will learn a lot more from this workshop. Carry the Vision restores the lives and communities through nonviolence education, creating a peaceful and compassionate world. Our goal is promote compassion, promote nonviolence. And for that, we have various programs. You can go to carrythevision.org and you will see various programs that are called Restore. There's a Restore for the kids, Restore for incarcerated people, Restore for people who serve the incarcerated people, Restore for communities, Restore for educational teachers, etc. Because everyone is stressed in this world, and everyone needs to take peace, become equanimity, and get peace in their mind. So it's a wonderful organization. It's been around since 2010. Actually, it was doing nonviolence conferences even before 2010 and 2006, 2007. So San Jose area had nonviolence conferences promoting nonviolence from Gandhi, Martin Luther King, Cesar Chavez, etc. So I welcome you all and urge you to look at carrythevision.org, and you can always volunteer to help us promote the nonviolence education. Thank you.
Again, thank you so much for Carry the Vision. They were our fiscal and platinum sponsors, and they've done an amazing job supporting us through this convention. So I'd just like to see, before we jump into our meditation session to kick off the morning, I would like to briefly discuss our own organization, STEM Enrichment Youth, which is hosting this convention. So we are a student-led nonprofit organization dedicated to inspiring middle and high schoolers to pursue STEM through engaging and motivating activities and resources. Our organization aspires to motivate students who have had limited contact with STEM to explore the field, as well as to provide guidance to passionate students seeking to become more involved in STEM. Our organization consists of roughly 70 incredibly driven high school and undergraduate students. This summer, we have created online classes for biology, physics, calculus, geometry and algebra, competition math, chemistry, and computer science. We host these classes through Google Classroom and create pre-recorded video lectures, assignments, activities, and problem sets. After Stemworld, STEMI will continue to organize similar conventions, including speaker events and STEM competitions. We are also starting a new set of live classes that will utilize virtual lab platforms and interactive activities. We will also be hosting a podcast where we interview STEM professionals about their work, career pathways, and the invaluable insight that they have to offer from their experiences. So if you're interested in joining us for more events, you should join the Stemworld Slack workspace or follow us on social media. You can also find all of our methods of communication organized on our link tree, which will be in the chat, in the subtitle, and later on in the Slack, on which you can also apply to join our organization. Lastly, before we start meditation, I would like to take the time to recognize everyone who contributed in organizing this awesome event. A special thank you to Natasha Lee, Yana Samoyolova, Brianna Doe, Claire Jin for creating all of our promotional material and posters for Stemworld. Thank you to Arjun Dixit and Sakshi for updating our website constantly on Stemworld. And thank you to Faith Han, Aryan Mathur, Arun, and Sophie for their amazing job with networking and connecting to schools and organizations that promoted Stemworld. Thank you to the rest of our events and operation team for Stemworld, including Devin Shaw, Yulia, Catherine Jo, Priyanka, Rahul, Vaibhav, Maddie, and Catherine Liu. And an incredible special thank you to our competition writers, Aniket Prasad, Catherine Liu, Harshdeep, Christian Kim, and Kailash. Now, before we proceed with many hours of exciting lectures and events, we're going to want to take the time to clear our minds through meditation. So please welcome Ann Fisher, if she's here, as she takes us away on our first meditation session. And if Ann is not here yet.
Does she have the same problem as I have with the link?
I shared her one link, so let's see. Okay, as to be expected, you know, there's a few technical issues. So we still have 15 minutes, so no worries. Meditation session should be only about 5 minutes. For now, if you want to ask any questions about Stemworld, you can put them in the chat or you can unmute yourself and just feel free to ask away if you have any questions about the day's events. So in terms of hours, someone asked how many hours will each day event last? Today we have events going from 8 a.m. to 7 p.m. You don't have to attend every event. We totally don't expect you to. But every sub event will be 30 minutes. So every speaker event, every networking session, every competition. Stemworld itself probably can't connect you with paid opportunities in STEM. STEM, if you choose to join our organization, we do have additional services. So we have several partnerships and our partners are very willing to help and support all of our members. Yeah. Awesome. Okay, Anne Fisher is here. And we were just going to start the meditation session. So whenever you're ready, if you could. Oh, Anne, you're muted.
Certainly. Hello, everyone. Thought we'd just take a few moments before we launch into this incredible weekend. If you just want to take a moment and get yourself comfortable, if you can, just put your feet flat on the ground, and your hands in a comfortable position. Just close your eyes. As we prepare ourselves for this time, let's just fully arrive in this moment. Let's take a deep breath in and a deep breath out. Just arrive in this moment with full expectancy as you open yourself to the limitless possibilities that will happen within and without. Letting go of all that which does not serve you. Just let it fall away like a backpack, just dropping to the ground, that you can fully arrive in this moment. Just naturally follow our breath. Allowing the mind to be clarified. And open to our own knowing. As you follow your breath, surrender into now. There's no place to go, there's no place to be, but right here, right now. As the thoughts come in, just let them move through. Just clearing the field, opening yourself to be receptive to this moment. And as you have arrived for yourself in this moment, may your mind be opened, your heart receptive, and be willing to explore the limitless possibilities of this day in your center, in the now. Let's take a deep breath in and out. And as you open your eyes, be ready, be open, and have an incredible day. Thank you all. It was nice to be with you.
Yeah, thank you so much, Anne. That was very refreshing. I hope everyone enjoyed that meditation session as well. So let's see. We have a few more minutes till our first speaker. So we'll just keep answering questions live. Okay, someone asked, do I have to participate for all sessions or as many as I like? How does the competition work? Is the competition a team event or individual? And will there be awards or is it just for fun? So again, you don't have to participate in any sessions that you don't want to. It's free to, give me a second. It's free to join for everybody. And I'm just going to post the Zoom link in the Slack workspace. Some people are having trouble getting in. Just so you guys should probably already know this, but for anyone watching, if you want to join the live Zoom, you can just go to our website, go to the Stemworld tab, and you'll see if you just click on any of the speakers' names, it will lead you to this Zoom link and you'll be able to enter our Zoom. So, okay. Okay, back to the question. So again, you don't have to participate in anything you don't want to. The competition, all the logistics for the competition are in the Stemworld Slack. And essentially, the competition will be 2 different Kahoots. And so we'll split everyone into 2 breakout rooms, and then the Kahoots will be running in parallel. And the top 9 total from the entire Kahoot will be receiving prizes. It's not a team event, it's individual. Yeah. And as for, can you access this after the convention actually happens? Yes, we'll save the live stream to our YouTube channel, so you can always go back and see any events that are live streamed. And just a disclaimer, most of our events will be live streamed, so you will be able to see them on YouTube, except for a few talks and all of our networking sessions, just because we don't want anyone participating in the networking sessions to feel like they're being recorded. Because those are just for connecting and getting to know everybody. Yeah. Okay, we have 2 more minutes, so you can just wait. Akshaya is here. So everything's ready. Let's see. Okay, last question, and then we're going to start with the bio panel. Let's see. Anyone? Okay, everyone seems pretty set. So we will start off with our first panel of the day. Awesome. So our biology panel will be a 2.5-hour long 5-speaker panel, as most of our other subjects go. And we'll be diving into the application of math and medicine, the importance of microbes in brain development, the use of bioinformatics in understanding microbial populations and complex human diseases, and the science behind memory forgetting. Before we get started, I would like to remind everyone of a few etiquette rules for all of our panels. So please refrain from disrupting the speaker while they're talking, of course. Make sure that you're muted at all times when they're talking. Please wait to ask any questions verbally until the Q&A session at the end of the speaker presentation. And please don't write anything discriminatory or offensive in the chat. If you do, and we see that you're consistently denying these stipulations, we will remove you from the Zoom session, in which case you probably won't be able to join again. So make sure that you're very conscientious of what you're saying. And so without further ado, Akshaya Anafragada is our very first biology speaker, and we're super excited for her to join us today. Akshaya is currently an MD PhD student at Johns Hopkins University, and she just earned her Master's of Science and BA in Applied Mathematics and Bioengineering at Harvard University. Today, she's going to be discussing the application of mathematics and engineering to solve problems in health and medicine. So everyone, please welcome Akshaya as she dives into the intersection of math and medicine throughout her college journey.
Hi, everyone. I'm really happy to be here today. I'll go ahead and share my screen so you can see some slides I prepared. So does that look okay to everyone? Perfect.
Yep.
So yeah, thank you so much for the lovely introduction, Diana. As Diana said, today I'll be talking with you about my work at the intersection of math and medicine. So just as a little bit about myself, while I was at Harvard, I was a research assistant at the School of Engineering and Applied Sciences and at the School of Public Health. And I was also the managing editor of the Harvard Political Review and did some advocacy work as part of our Women in Computer Science group. And now I'm a first-year MD-PhD student at the Johns Hopkins University School of Medicine. And I'll be sharing some examples from my research first, and then trying to have a conversation with you about this. And I'm happy to take questions at the end. So to get started, one of the things that I'm really interested in is how mathematical models and machine learning can be used to answer big questions. And when I say big questions, I'm talking about sort of systemic macro level questions, things that affect global health, policy, and clinical systems. And I think when looking at these kinds of questions, taking a mathematical approach has several advantages. First, it allows you to use and learn from real-world data. So in practice, data is noisy. It's often messy, and different types of data are collected in different ways and at different intervals. And computational approaches are really good at finding ways to make sense from all this noise. Modeling also lets you try different scenarios quickly and efficiently, in circumstances where a controlled experiment may be impossible. And it also lets you capture those really complex dynamics in order to design and evaluate tailored interventions. So these are some of the reasons that I think mathematical modeling has so much potential in medical applications. Now, another thing that I'm interested in is model interpretation, which is the idea that once we have a model or an algorithm, we need to understand what it does and how and why it makes the decision that it does. So these memes that I have here, they're kind of jokes, but I think they're also representative of this broader idea that machine learning and mathematical models more generally are black boxes, and that we can't possibly know what happens inside of them. So we have data and we give it to the model, and then the model tells us what to do, and we do it blindly. But this is actually really dangerous. It allows us to create models that reflect human errors and biases, and it prevents us from gaining new knowledge about what kinds of factors matter in a system. So something that I look at a lot in my research is interpreting models after they're built and trying to use this to gain insights into medical practice. So with those general principles in mind, I'd like to share 3 examples from my own research where I've built, evaluated, and interpreted mathematical models with the goal of answering some kind of medical or clinical question. So for my first example, I'd like to talk with you about work I did with the Golden Lab at Harvard to mathematically model poultry vaccination programs and design interventions designed to enable sustainable food sources in Madagascar. So the summer after my first year of college, I spent time in Madagascar working with the Golden Lab. And we were interested in sustainable food sources for really remote communities in Madagascar that are reliant on hunting wild meats in the rainforest, things like bats and lemurs. In these communities, there are already staggeringly high rates of malnutrition. As many as 50% of children under 5 are nutritionally stunted. And as the rainforest declines, it's only exacerbating this food insecurity. Moreover, consuming wild meats poses a risk for the transmission of zoonotic diseases. Now, chickens are a potential domesticated alternative to wild meats. However, they are highly susceptible to Newcastle disease, which is a fatal respiratory disease endemic to Madagascar that kills large proportions of flocks. So this makes chicken an unreliable food source. People don't want to invest money and resources in raising chickens if they're just going to die before they can be eaten or sold. Now, vaccinating chickens against Newcastle disease is possible, but mounting a vaccination campaign is really hard in the context of some of the most remote locations on Earth, without electricity and reliable cold chains. Therefore, we turned to data on chicken populations to try to model possible interventions. So in my work, I used data that field teams from our lab had been collecting on Malagasy chicken dynamics for several years. So things like births and deaths and purchases and sales and consumption, which are all ways that chickens enter and exit the population. And we also use knowledge of the vaccine, for example, that it must be administered 3 times a year to maintain immunity. And then I used all these different data to build a system of differential equations to model chicken populations in Madagascar under various vaccination regimes. And I solved this system numerically. And then I interpreted the solutions with respect to 2 different quantification metrics. So the first was population doubling, which is a measure of population growth. In Madagascar, human populations are increasing rapidly, and food security pressures are increasing as well. Therefore, to meet this demand, it's important that vaccinations suppress chicken deaths enough that flock sizes can grow. And we found that this type of population doubling was readily possible, with 40% vaccination coverage over 5 or more years. But the second factor we looked at was herd immunity, which is the idea that once a certain proportion of a population is immune, then disease outbreaks won't really spread. So we know from other studies that this metric is around 85% for Newcastle disease. But it turns out that triennial vaccination can't achieve this because of the time scale in which chickens are born and die. It just might not be possible to use vaccination to maintain this pool of immune population over long time periods. So this modeling gave us important insights into how vaccination could be used to stabilize chickens as a sustainable food source. Now, one thing that I think is really cool is that this vaccination program is currently being used in several Malagasy communities. So that's an example of how modeling insights can be translated into actual interventions. So now I'll switch tracks completely because I want to give sort of a different vignette about my research. And I want to talk about work I did at Baylor College of Medicine and Texas Children's Hospital to detect child abuse in the electronic medical record. So non-accidental trauma, or NAT, is a clinical euphemism for child abuse. And it turns out that child abuse can be difficult to detect and diagnose for several reasons. First of all, the patients are usually young and nonverbal, and given the situation, you may not be able to rely on their caretakers to be accurate conveyors of information. The clinical presentations are also just confusing. So to give you an example, this could be a fairly typical clinical picture. You have an infant with a skull fracture, cerebral hemorrhage, and localized bruising. The mother reports that the infant was on the bed. She turned her back, then heard a crash, and found the baby on the floor crying. This immediately begs the question, is the injury mechanism described consistent with the injuries the child presents with? If not, this might be physical abuse, and now there are questions surrounding who was responsible, whether the story that the adults are telling is consistent, and whether CPS should be involved. Now, if the injury mechanism is consistent with the injuries, you still have questions over whether this was truly an accident or whether the parents failed to monitor the child adequately, which could be neglect. So regardless of the determination, there are still questions surrounding whether this is a pattern of injuries, whether they're complicating social circumstances, and how serious the injury is. So this seems really complicated. But now add in that smaller community hospitals rarely have physicians specifically trained in child protection, and that children may see several different healthcare providers across numerous clinical sites and time points. And you have a pretty difficult problem. But these challenges are compounded by the fact that much of the information needed to come to a decision is in the electronic medical record, which is largely free text notes. So it's hard to automate its processing. And the stakes are incredibly high. Unlike in many medical applications, both false positives and false negatives are really bad. So to give an example, in some medical applications, false positives aren't great, but they can be investigated by further testing. However, with NAT, false positives can lead to traumatic separations from caring families. So it's really important to get it right quickly, effectively, and the very first time that there's a suspicion of NAT. Therefore, we asked the question, how can we find ways to process the massive amounts of text in the medical record to flag possible cases of abuse for physician follow-up? So here we use natural language processing to numerically encode the free text in the medical record, and then we pass these encodings to neural networks that were able to predict the probability that a specific patient record corresponded to abuse. So we tried 3 different natural language processing techniques. In Bag of Words, we take all the words in the record, scramble them up, and assign values to them. So there could be tens of thousands of words in the record, and we consider all of them. In contrast, for rules-based, we consulted with expert physicians to identify phrases in the medical record that could be important, and then we scored each record for the presence or absence of these phrases. Finally, in word embeddings, we used a pre-trained structured text representation to encode the medical records, kind of like using a translation from another language to make sense of this specific record. Finally, when we passed these encodings to the neural networks, we had mixed success in making predictions. Ultimately, the encoding strategy that was most successful was bag of words. And when used with a neural network, it had accuracy and AUC of over 90%. So just as a quick aside, AUC is a measure of the trade-offs between false positives and false negatives, and higher values indicate stronger performance. So this was really great results. We had a model that made good predictions. However, we didn't know a lot about how the model was making predictions, which is concerning, because for something like child abuse detection that is known to have racial and socioeconomic biases, it's important that our models aren't learning those biases. So we used something called a saliency analysis to look at the most important model inputs. And we found that they were highly relevant radiological words, things like fracture, swelling, or head, as well as the phrase history of domestic violence. Now, all of these are documented in the scientific literature to be signs of abuse or correlated with abuse. And importantly, they aren't racial or socioeconomic factors that reflect an underlying bias in diagnosis. So this is a really important point, I think. By using model interpretation, after construction, we were able to gain insights into what parts of the medical record were important to the detection of child abuse, and make sure that what we were paying attention to made sense. So now, for the final example I'll give today, I want to talk about my recent senior thesis work with the Golden Lab and Professor Milind Tambe at Harvard, where I developed models to predict malaria and anemia in remote areas of Madagascar. So just to set some background, these are some pictures of the communities in Madagascar where I was working. Unfortunately, these are some of the poorest communities on Earth with really severe health challenges. There's very high malnutrition, with about 50% of children under 5 being growth stunted. And these nutritional deficiencies contribute to staggeringly high rates of anemia, while malaria prevalence is also very high and concentrated in these remote communities. These high disease burdens exist against a background of low access to healthcare, with over 50% of the population living more than 5 kilometers from a health center. Unfortunately, that last factor makes diagnosing malaria and anemia really difficult, since both of these diseases require blood samples to diagnose. So for folks far from clinics with the needed staff and equipment, they can be almost impossible to diagnose. An additional complication is that in areas where these diseases are prevalent, the coincidence of the two is common. This coincidence is especially lethal. By itself, anemia causes a decrease in red blood cells, and untreated malaria leads red blood cells to burst and die. So the combination of the two diseases can cause a precipitous drop in red blood cells that has a fatality rate of about 50%. Therefore, it's really important to find ways to diagnose malaria and anemia using non-clinical variables so that we can figure out who needs treatment at a hospital urgently and who's at risk for these diseases. So in my thesis, I used a data set of over 5,000 individuals from 24 sites across Madagascar that came from 4 different regions with really different environments. The people were both male and female and of all ages, and I also had over 1,000 different non-clinical variables. So things relating to diet and nutrition, economic status, demographics, income, etc. And what I did was use these variables that can be easily assessed outside of a clinic to build predictive models of disease status. So I built 3 types of models. The first used a technique called principal component analysis that transforms the data into an alternative space and then tries to look at which variables are responsible for variance in disease status. This didn't work very well because, unsurprisingly, a lot of variables contributed to this variance rather than just a small set. The second type of model was logistic regression, which tries to fit a logistic function to the data and then approximate the relationship. And this was overall pretty accurate, but it missed a lot of individuals with disease. So it was good at predicting negative individuals, but not good at predicting positive individuals, which is a problem for a medical application. The third type of model I tried was neural networks, which are a type of deep learning model that are really good at capturing nonlinear relationships. And these models were really good. They were 80 to 90% accurate and identified positive patients effectively. So this is the kind of model you could try to deploy in the field. Next, I tried to use statistical tools to reverse the models and figure out which variables were important to disease status classification, with the idea that these variables could become targets for public health interventions. And what I found is that outside of a few obvious biological factors surrounding age and sex, the majority of important variables had to do with poverty. Limited access to food, nutrients, safe water, and sanitation, as well as gender inequality, seemed to be big drivers of disease. In contrast, wealth and things that go with it were protective factors. And I think this is an interesting conclusion, because it tells us that I took the most sophisticated mathematical models in the world and threw them at this problem. And what they came back with was essentially that poverty alleviation and development work may be the most useful thing we can do to prevent malaria and anemia. So I see these results as a call for broad, multi-sector development programs that can help lift communities out of poverty, consequently reducing disease burden. But the other thing that I think is cool is that I came to this conclusion by building mathematical models and then turning them on their head and interpreting them. So I think this type of interpretation is a powerful technique for understanding complex phenomenon that seem opaque at first. So now that's an overview of my research. And now I'll try to offer some brief advice for others interested in this field. So to be honest, I feel a little silly trying to offer advice here. I'm not nearly as accomplished as many of the other speakers this weekend who are like distinguished professors and experts in their field. So it's a real honor to be speaking alongside them. But the event organizers did ask that I include this section. So I'm going to do my best to offer sort of a student's perspective. So I first want to talk a little bit about getting involved in research as a young student, particularly in high school or even early in college, when you have a lot of interest and enthusiasm, but may not have a ton of technical mathematical skills. So first of all, getting data can be a big challenge. You probably saw from the examples I gave, almost all of the data I used to build models was data members of the labs I worked in collected in the field, or it was data from the hospital I worked. And these types of data tend to be confidential. So I think that can sometimes be a barrier to working this field unless you're part of a big academic enterprise that has the data you need. But good news, not all data is like this. And I think relying on publicly available data sets can be great for learning and exploration. One of the sources I've used in the past is Kaggle, which is a website that has a ton of interesting data sets freely available. So there's currently several on COVID-19, and there are cancer imaging data sets and all kinds of other interesting things. Another big piece of advice I have is to learn to code. You can start doing this today, and it's one of those skills that takes time to build and requires practice. But if you can code effectively and efficiently, it makes it that much easier for you to get involved in projects when you find them. So I'd recommend Python, which has a ton of free open source libraries and frameworks to support almost anything in the data science and mathematical modeling realm. I'd also recommend looking for ways to enter science fairs and competitions. So when I was in high school, I entered both the Science and Engineering Fair of Houston and the Siemens competitions, and I had a lot of fun. Even if you don't discover anything earth-shaking, and I certainly didn't, they do give you experience in planning and executing a research project. Finally, I'd look into summer research programs at universities that allow high school students to work in university labs as assistants during the summer. These can be a great way to learn and start getting experience. Now, as far as more personal advice, I think one thing that served me well is saying yes to anything that comes along. So you probably saw that I've worked on a lot of different topics and in very different settings, from the field in Madagascar to an academic hospital. And I think that these have all been valuable opportunities, and they've all sort of built on each other and helped me develop a portfolio of work. The way I see it, you never know what you'll like until you try it, and connections tend to sort of snowball. So the more involved and enthusiastic you are, the more chances you get. And you never know which one of those will be the one you fall in love with. So on this note, I'd also stress the importance of seeking out good mentors and positive teams. In all the projects I talked about, I've been really fortunate to learn from incredible professors and to work alongside colleagues who I've had a lot of fun doing science with. And I think this is key. Mentors and colleagues can help you get projects done, for sure, but they can also help you plan out your career and be successful. And they can really make or break your research experience. So I think it's important to be intentional in finding supportive, knowledgeable mentors and lab communities. And I also think it's pretty important to collaborate across disciplines and try to draw on the strengths and tools of different fields. Whether you're looking at engineering or math or medicine, I think bringing them together can be really valuable. And the other big piece of advice I have is to always keep doors open. The best way I know to do this is to just, as a principal, do your best work on every project you're on. I think it's unrealistic to assume that you'll enjoy everything you work on, but the way I see it, doing good work is sort of a muscle, and you have to exercise it to make it stronger. So no matter what, if you do the best you can at anything you're working on now, learn as much as you can and try to go above and beyond wherever you can. This sets you up to be able to do exactly what you want in the future. And then I also think it's important to make plans and set goals and just constantly be evaluating and reprioritizing. So I like to set, short-term goals for myself as well as medium and longer-term goals that concern my academic plans, my career choices, and where I want to be in the next week or the next year or even the next decade. So I think these are some strategies that work for me, and I hope some of them are helpful for you too. And then finally, I would be remiss if I didn't thank and acknowledge the tremendous number of people who've contributed to this research alongside me and served as mentors and colleagues in doing this work. I'd like to offer special acknowledgement to my main mentors on these projects, Professor Chris Golden at the Harvard School of Public Health, Professor Zbigniew Starosolski at Baylor College of Medicine, and Professor Milind Thambe at the Harvard School of Engineering and Applied Sciences. I've also listed the key publications and abstracts that I've authored on these projects in case anyone's interested in reading more. And now I would be happy to answer any questions.
Awesome, thank you so much, Akshaya. Let's see what everyone has to ask. All right, while we're waiting for anyone to come up with questions, I have a few of my own. So these are less related to your exact work, but more about your professional pathway. What was your favorite aspect of attending Harvard as an undergrad?
Yeah, absolutely. I think this is probably true of any college, but I just enjoyed living and learning with peers who are interested in a lot of things that I was interested in too. And I think I had really great opportunities just to work on exactly what I wanted to work on and the way I wanted to work on with the people that I was excited to be with. And I think that sort of communal aspect is a really favorable thing that a lot of people enjoy about college, and I did too.
For sure. Let's see. YouTube, anybody?
Yeah, so I can kind of answer these two questions together about the 90% success rate and the question about implementing ML algorithms. So yeah, a 90% accuracy rate does mean that 10% of these are incorrect. And I think that is a problem. But one thing that I do like to think about is that this is not one of those things that we're great at doing without computers. So physicians making these diagnoses are only right about 75–80% of the time on their first guess. There's usually quite extensive follow-up. So if on our first pass we can get to 90%, that's a good opportunity to then involve physicians in further workup of the patient. So I see this more as an early detection flag than the final, end of story, we are deciding what's happening. And then the question about getting child abuse detectors implemented in hospitals. So no, this is not yet implemented in hospitals. And in general, I think there are a lot of interesting regulatory questions around how machine learning algorithms will be used in a hospital environment, because right now there's a lot of great research happening on them. But as you probably know, there are a lot of laws concerning what you can do in a hospital. There's privacy laws, there's the FDA that regulates products that are being used in medical settings. And it's a pretty big question how these can be used. And I think right now, a lot of people are finding out that translating research to clinical practice can be a tough thing. And so we're continuing to work on this and try to improve it and hopefully get it used in the hospital. But I think that the main steps there really are doing the research, getting it published, and extensively validating it before you really try to make it a commercial product. Okay, There's a question about if I can recommend sources, books, advice on improving science skills. So beyond the sort of general advice to look for data sets to start learning to code, I'm not sure I have specific books or sources, but I would say that there are a lot of freely available resources on the internet. I think Code Academy comes to mind if you want to start learning Python. And then I think my general advice would be to just start playing around with it as quickly as you can. So you can spend a lot of time reading textbooks. And I think that reading textbooks can be really valuable. But where I really found like specific books to be useful is once I know what like the specific challenge I have is, and then I go to look up what can I do with it. I don't know that for myself, reading books cover to cover before starting has been extremely useful. But that's, just kind of my thought. When is a good time to start joining research teams in high school? And where did I learn to create all the different types of models? Okay, so joining research teams, I would say now. Like if there's a research team that you're interested in and you want to join it, join it. If you hate it, you can always find a way to stop doing it after. You don't want to leave people in the lurch, but it's always fine to quit things that you don't enjoy. But if you do enjoy it, why not start your future career today? So that's kind of my advice on that. And then creating different types of models. I think some from coursework, but a lot of it is also reading the literature. So once I know this is the medical problem I want to go after, I start reading scientific papers and trying to see what's already been done and then building from there. And then maybe I go read some mathematical and computer science papers to figure out how to implement the model. So I think it's pretty iterative. I definitely didn't learn all of this from my coursework before I started doing research. Okay, what sort of research can we do without needing to go to labs in public places? Research that we can do online from the safety of our home with the guidance of a mentor online. Yeah, so I think that this kind of computational research is actually a great example of like COVID-proof research. You don't have to go outside, you can sit inside with your laptop. And I think finding those publicly available data sets and just diving in is a great place to start. And as far as mentors online, I think anyone who would have been a mentor in person pre-COVID can be a mentor online. You just need to chat with them, and like everything doesn't translate perfectly to Zoom, but I think a lot of things can be done over video chat or phone calls that you might have once done in person. Okay, how much do college professors assist you in finding research opportunities like yours? Yeah, so I think that varies quite a bit. I definitely don't think that there are college professors who are just, taking out ads in the newspaper saying, I need a student to come write the following code and then it will be done and the research project will be published. But I do think that most professors have some idea of what they want students in their group to work on. They're usually not going to say oh, just come on in, I'll give you money and time and resources, do whatever you want. So I would say the balance is usually the professor will come up with the big idea, but you'll come up with the implementation steps. They probably won't tell you what model to use. They'll just tell you, we need to find a model that answers questions in this realm. And then you'll hone in on the question and you'll hone in on the techniques. Okay, what is the difference between Python and R for data analysis? Which one is better? So as far as differences, I think the big thing is that R is a statistical language, and there are toolkits that are being built to do more modeling approaches, but fundamentally R was originally used for data science statistics type things, whereas Python is just a multi-purpose scripting language. And again, now there are libraries being built specifically for modeling and machine learning and things like that. But I think they both just started out with very different purposes, though you can now do kind of the same thing as neither one. And in my opinion, I guess I would say Python, but a lot of that is I learned Python first and I know how to use Python. Whereas when I see R code, I have to think how to use it. So it just kind of, I prefer Python, but I think it's open to your preference. And then I do use MATLAB a little bit. I think MATLAB really shines in signal processing and in some image interpretation tasks, whereas Python is a little lighter weight and just easier to use when I'm trying to do some exploratory data analysis.
It is actually 9 a.m., so maybe we can have one last question if anyone has any lingering thoughts. Oh.
Okay, I'll try to answer those last two together. So prerequisites, it really depends. You just need to talk to the professor or look into the opportunity. But in general, I think even if you don't have all the prereqs, if you have some of them and you're enthusiastic about getting the other ones, I don't think it's going to be a huge barrier. And then advice I would give to someone considering the MD PhD path and recommend to become a competitive student. Okay, the only advice I really have if you're considering it is to go for it. Like, I mean, I don't know, I'm only a year into it, but it seems pretty okay so far. And I think that it's really cool to be thinking that one day I'm going to be both treating patients and finding ways to, come up with better science to help those same patients better. So it's, I think it's a good career. And what I recommend students to do to become a competitive student. Yeah, so I think those two pieces of advice that I had about just saying yes to opportunities that come your way and doing the best you can at everything you touch are kind of the two I have. Like, it takes practice to be good at things. So practice being good at things. And then that way, when the thing you really, really want to make your career out of comes along, you'll kind of know what it takes to be great at it. So that's kind of my advice. But I wish you all best of luck in your own paths to becoming students and researchers.
All right, thank you so much, Akshaya. I hope everyone really enjoyed learning about Akshaya's application of math in medicine and how she was using mathematical modeling to design vaccine interventions and, help preventively diagnose child abuse. Our next speaker is Dr. Helen Wong. She's currently a postdoc at UCLA, studying the role of the maternal microbiome in fetal brain development. Dr. Wong earned her BS in neuroscience from the University of California, Los Angeles, UCLA, and she continued her graduate education at UCLA, earning her PhD in molecular, cellular, and integrative physiology under the mentorship of Dr. Nicholas Brecha. Without further ado, everyone, please welcome Dr. Helen Wong as she dives into the importance of the microbiome in development.
Hi, everyone. Thank you for good morning, first of all. And I appreciate you guys spending your Saturday morning with me. I'm going to try and figure out how to share screen. I'm still getting used to this whole Zoom meeting. Let's see. Oops. All right. Thank you, Diana, for organizing this STEM Enrichment Youth Conference. It's amazing that you're able to put this together. I saw in the chat that it just started this year and you just started in June or something like that. So just in a few months, that's amazing. And so I am very excited to be able to share with you guys some of the work that I've been doing and what I can impart on you at this stage of my career. And so just as a title, most of it's going to be focused on the gut microbiome, brain and behavior and health and disease, but I kind of want to inject some previous work that I've been working on as a graduate student. Okay, so like Diana was just saying, I did my undergrad at UCLA. I did it in neuroscience. I didn't actually think that I was going to be doing neuroscience. I really did enjoy science in high school, but I didn't know what I wanted to do once I got into college. So if you're still figuring it out now, that is, I think, absolutely okay. I know lots of people that changed majors in the middle of their college and figured out, what they wanted to do later on. I also did my graduate career at UCLA in MCIP, and that was with Dr. Nick Brecha, and we did, I'll explain that a little bit later about retinal visual processing. And then finally, I did my, I'm doing my postdoc right now at UCLA with Dr. Oops, sorry, what's going on here? Okay, sorry. With Elaine Shao at UCLA, and that's where the bulk of my work is right now, is looking at microbiome on host behavior in her lab. And in the middle of all of this, I also had a son, and here is his cute face, and very proud and happy to have a healthy baby boy during these COVID times. So that's what you can look forward to if you would like to have children in the future. And so just to go over really briefly, some of my graduate work was looking at neural cells in the back of the eye. And these are called retinal cells, and they process visual information before it goes to the brain. So all that light and shadow and visual processing begins in the retina. And my work really focused in on how these amacrine cells communicated with use a laser pointer, communicated with these retinal ganglion cells. And I used different techniques, including looking at morphology, using confocal microscopy. I looked at how two different cell types communicated with each other, formed these contacts, and I also looked at their functional output. So taking an electrode, sticking it into these cells, and then recording the electrical activity. And you can see these little spikes. These little spikes indicate that there's some firing and communication happening. And if you take a drug or you stop the communication, you lose those spikes from happening. And that's why you get these decreased spikes. And so most of my graduate work was really looking at this model of neural communication in the retina. And when I finished my graduate work, I was very interested in understanding whether our body was specifically isolated in how it functioned. And it turns out we're not isolated. What makes us us is actually very interesting. We co-evolved with trillions of microorganisms. These include bacteria, virus, fungi, and that these microorganisms inhabit our body, and they've co-evolved with us, and they are in our body and on our body, and in different locations of our body, there are they're all very unique in terms of what are the microorganisms that are present, and they have a very variable composition. Just by looking at these micrographs that are about 3,000 fold magnification, you can see that these little microorganisms that aren't really visible to the human eye are very different from location to location. And so we have only really scratched the surface in this field about understanding what each of these little bugs are doing and how they could be potentially influencing our host physiology. And so one of the kind of cool projects that have come out about the microbiome is the Human Microbiome Project, which was a National Institutes of Health initiative that started around 2007 to 2008. And what they essentially are trying to do in this Human Microbiome Project is to profile all the different types of microbiome in healthy people, as well as people that have a disease model like IBD or diabetes, or even women that are pregnant. They all want to this project essentially wants to do is profile all the different microbiomes. And some kind of cool facts that came out of this project is that microbes, in terms of their numbers of cells, actually outnumber the cells that make us from 1.3 to 1. And if we look at the genetic material from microbes versus human genetic material, they actually outnumber us 150 times. And if we think about all the different types of microbial species that are present, there are over 10,000 different ones on our bodies. And what is really interesting is that a lot of it is accumulated in our GI tract, and 95% of the microbiota is in our gut. And if you take all that microbial mass from our body and put it into a container and try to measure its weight, it's around 2 to 6 pounds. And just to give you some perspective about what that means, our brain is about 2.6 pounds. Our heart is about 0.7 pounds. So it's very interesting that something that we never really thought was us is actually either the same or more than many of our other organs in terms of weight. And so based off of this information, I became really interested in how these little bugs could essentially be affecting our body. And research has been ramping up, and scientists have kind of gone into these different avenues and try to understand what the microbiota is doing in terms of affecting metabolism, how we process food, or the immune system, how we can fight off different types of infections, drug metabolism. So how quickly or how efficacious our drugs are being used in our body is dependent on the microbiota. Intestinal function. So if someone has ever had GI disorders, intestinal motility issues, this could be due to the imbalance in the microbiota. Bone homeostasis, development of the bone has been implicated in microbiota. And then what I'll really focus on today is how the microbiota is affecting our brain and behavior. And so this idea of microorganisms affecting host behavior is not new. It's actually been studied in many different organisms. And so this Ophiocordyceps unilaterus is a fungi. And essentially what it does is it hijacks the ant's body to actually cause the ant to climb to different levels of a plant so that it could get more or less sunlight to grow. Another example is the rabies virus. It can attack, most commonly known in dogs, and really causes aggressive behavior. And the idea of causing aggressive behavior is to actually cause these dogs to bite other dogs and spread the rabies virus. So again, it's to promote their development or their proliferation. We have Toxoplasma gondii. I'm not sure if people have heard of this, but it's more commonly known as toxo. What scientists have found is that this particular Toxoplasma gondii is able to infect mice. And what they do is it causes the mice to lose aversions to the cat, and it will lose aversion to cat pee, for example, and it'll cause the mouse to actually go towards the cat. The cat will eat the mouse, which will allow for this Toxoplasma gondii to proliferate, because it only really survives or can live in the feline gut. And then finally, there's the Salmonella enterica. It also affects our feeding behavior, and ultimately the goal is to proliferate inside the human gut. And so my work is going to be focused on not necessarily all those other microorganisms, but the gut microbiome in particular and how it can affect host behavior. So this is just a pictograph of a intestine that's been stained with different markers that are localized to either the mucosa, some intestinal cells, and then this is the lumen over here, and all these different colors over here are different microbes or bacteria that are present. Oh, sorry, am I drawing? It's weird. Okay, so, and then the idea is that we are interested in how those bacterial products or these different bacteria could be communicating with the brain. And scientists have begun parsing the role of direct communication through the vagus nerve. So there's this particular nerve that goes from the intestine all the way up to the brainstem. There's also the idea that there are neurotransmitters, hormones, or small molecules that come from these microbes or the host that go through the circulatory system and communicate with the brain. And then finally, there's the idea of there's this communication between these microbes and the intestinal immune system that can also be creating MAMPs or metabolites that communicates with the brain. And so we are actually really most interested in the circulatory system pathway, but there are tons of research in all of the 3 different linking pathways. And how do we study this? It's actually really hard to do it in humans because you can't really isolate specific bugs and then take out the brain of a human and then do all these behavior stuff on them. So a lot of the work that we do is using animals, and we use a technique called notobiotics, which is, it means known life. And what we do is we use mice and we put them in these isolators or these bubbles. And in these bubbles, they are completely sterile. They lack all types of microbes. And so this allows us to really control for what kinds of bacteria or whatever microorganisms we're studying and look at what their effects are. And so essentially what we have are we take these mice and we can either make them germ-free, which is in these isolators. Alternatively, we have this technique using antibiotics, which will clear all or most of the bacteria in the gut. We also have specific pathogen-free group, which is essentially our control group. They have the full microbiome, nothing's been done to them. They're completely normal in the sense that they have a full microbiota. And then finally, we have the specific bacteria. So we colonize these mice with specific bacteria that we're interested in and trying to understand what their role could be. And so previous research from scientists have already begun understanding what the role of the microbiota is doing in animal brains. And so, for example, in germ-free mice, there are decreased expression of these key brain genes like NGF and BDNF. As you can see, comparing a control animal with a germ-free animal, you see there's a significant decrease in the amount of bright spots. Similarly, there's an altered stress response in these germ-free mice. So usually when animals are stressed, they release these different types of molecules, but then in the germ-free mice, they have this altered level of these molecules. There's also anatomical changes. Germ-free mice have these different dendrite processes and microglia formation. And also there is a altered blood-brain barrier integrity. So there's tends to be a more a leaky brain where other molecules can easily enter the brain in these germ-free mice. And not only is there these sort of anatomical changes, but there's also been behavioral evidence suggesting that the microbiota has a major influence on how animals will act. And so some tests that scientists can use is this social interaction test where they put a mice into a 3-chamber plexiglass box. They put them in the center, and then they will either time how long how long they spent in the chamber with the mouse that is a novel mouse or an inanimate object, or how many times they enter one versus the other. And typically, mice or rats are very social animals, so they'll tend to go towards the object or the other mouse. And what they found was, yes, in a control animal, they spent a lot of time with the novel animal. But then if you deplete the animal of all of its microbes, it becomes less social. It actually spends more time with the inanimate side versus with the animal. But if you take that germ-free animal and you gave it back some bacteria, it actually goes, normalizes itself. It comes back and interacts with the novel animal. So it's very interesting how these bacteria are now able to influence different behaviors. And then here's another example of another behavior, which is an open field test. You put the mouse in the center and you see how much time it spends in the center versus on the outside. And typically rodents like to spend their time on the outside versus in the center, because center typically means danger. And so interestingly, the control spends a little bit of time in the center, but then the germ-free one spends a lot of time in the center. So they have this change where they are not as scared. And so they spend a lot more time in the center compared to the outside. And here's just some recent evidence suggesting that the microbiome is not only affecting baseline behavior or baseline anatomy, but it also has a very interesting role in modulating disease phenotype, in particular neurological diseases such as Parkinson's. And in this paper, they look at germ-free animals in an alpha-synuclein Parkinson mouse model. And they show that this animal is able to, it has an altered Parkinson's response or time to cross when you deplete the microbiome. Similarly, there's another paper looking at ALS, and they do a bunch of behavioral readouts in the animals with the ALS transgenic mouse line. They actually have an altered response when you deplete the microbiome. And so this is all really interesting in highlighting what we want to do in our research group. And that is, again, focused on brain function and behavior. We wanted to take a step back and we wanted to understand how the microbiota impacts brain development. So as an infant or a prenatal animal, how is the microbiota affecting brain development? And then what is the role of the microbiota in seizure development, in a disease model of seizure? And so to jump in on the microbiota side of how it affects maternal development or offspring development from the maternal environment, previous work by Elaine Shao, she looked at how different autistic-like behavior, such as stereotype behavior using marble burying, when the moms were exposed to an infection, which is a mouse model of autism, the offsprings like to bury the marbles a lot more. But if you gave the mom back this Bacteroides fragilis, it actually reduced the amount of burying. Similarly, the amount of communication is different. So in the mouse model of where they have the maternal immune activation, they have the offsprings have reduced number of calls, the call times are reduced, but then if you give them back this Bacteroides fragilis, they have these normalized communication in these offspring. And so our work is focused in on what about the maternal microbiota is affecting the fetus. And so we asked some key questions. What does the physiological maternal gut do for a brain? And what is the mechanism? And so what we did was look at the gene expression. Essentially, this is just a heat map looking at up or down-regulated genes. And we found a lot of these key genes were related to axon development. And so when we take embryonic brains from mice that, or their moms were had a complete microbiota or a depleted microbiota, we saw that the fetuses had reduced axons compared to in the maternal microbiota that's depleted compared to a full microbiome. And not only do we see brain differences, we saw behavioral differences. And some of the behaviors we saw were these tactile sensory behavior. These microbiota depleted offsprings have a longer time to respond to these stimuli. And this suggests that they have this sort of hyposensitivity in their peripheral response to sensory stimuli. And just as kind of using, this is a random forest analysis using machine learning, which is kind of something that Akshaya touched upon. Essentially what they did was they looked at these small molecules in the maternal microbiota that's produced from these microbes and try to see whether or not these little small molecules could affect brain development. And so taking these small molecules and pairing them with different microbiotas, we're able to predict with 87.5% accuracy that these bugs, or sorry, these little metabolites could be associated with a particular maternal microbiome. And what's really cool is that we found some that were decreased in the microbiota depleted animals compared to the non-microbiota depleted animals. And that if we gave those small molecules back into the mom and looked at the embryo brains, we can see these axons grow right back out. And not only do we see the sort of anatomical changes, but we see the behavioral restorations where they have normalized tactile sensory behavior. And so this is just in summary that the maternal microbiome produces these small molecules that affect growth of these axons. They get to the brain, and then it also affects behavior. Let me just check how we're doing on time. I think I have a few more minutes here. And just to kind of touch on the second story, which is how we looked at epilepsy. So epilepsy is a neurological condition based off of disturbances in electrical activity. And the refractory seizures that occur in children are really uncontrollable, and they don't really respond to anti-seizure medication. And it's a worldwide burden, with 65 million people around the world having epilepsy, and 1 out of 3 have uncontrollable seizures that don't respond to treatment. A kind of not so new treatment, but has its downsides, is the ketogenic diet, which is a high-fat, low-carb diet. It's been used to treat infantile spasms, Dravet syndrome, GLUT1 deficiency, and people have used it, or scientists have used it for infants using a gastronomy tube. However, the ketogenic diet, if anyone has ever tried the ketogenic diet, it's just basically Crisco. It's very harsh. It doesn't taste good. I mean, you can imagine as a child trying to take ketogenic diet every day is a terrible way of going. And so this kind of motivated us to kind of understand what the ketogenic diet is actually doing. Could it be doing this through modifying the microbiota? And then could we then harness this to develop something that could be more efficacious in treating childhood epilepsy without using the diet? And so we used a mouse model of 6 Hertz psychomotor seizure in collaboration with a graduate student in our lab, Christine Olson. And what we show is that the ketogenic diet is effectively reducing the seizure threshold. So it takes a higher stimulus to induce a seizure when you are taking the ketogenic diet. The ketogenic diet significantly changes the microbiome. You see, this is the starting microbiome over here. And then through the different diets, a ketogenic diet versus a controlled diet, you have very different microbiomes. And what we found was that two particular bacteria are sufficient for carrying out this ketogenic diet effect. And one of those is Akkermansia acidophila, and the second one is Parabacteroides. If we took those two bacteria and colonized the animals with them and then did the seizure testing, we saw that these bacteria effectively colonized the gut, and that it also increases their seizure threshold, which is really cool. We were able to find that they don't work independently of each other. So if you just gave them Parabacteroides or just Akkermansia, it doesn't work. You have to give them two together. And if you gave them a random bacteria like a Bacteroides, it's not as effective. We used finally another model, which is a KCNA, which is an actually occurring transgenic model of seizures. And we get these really, you do EEGs, which is electrical activity in the brain. We record that. And what we found is that in these animals, they typically have these really crazy seizures. They spontaneously occur, and you can see here that there's a lot of uncontrolled brain activity that causes them to jump around. When we give these KCNA mice back some of this Akkermansia Parabacteroides, we're able to reduce the seizures and the time that they have seizures. So this is really exciting for us that we were able to find something so effective. And we wanted to figure out how it was doing this. And so just very briefly, we again looked at small molecules that were produced in either the blood, or it was produced in the gut that makes its ways into the blood, or is still in the gut. And then we looked at the actual GABA to glutamate ratio in the animals. And we found that Akkermansia Parabacteroides actually normalizes this GABA to glutamate ratio, allowing for normal brain activity. And just in brief, this ketogenic diet regulates the gut. It regulates these specific metabolites in both the colon and in the blood, and essentially increases this GABA glutamate ratio to cause seizure protection. And so hopefully you guys are interested and amazed as much as I am about this gut microbiome and how it could affect not only the developing brain, but a diseased brain. And our goal is to continue to understand how we can harness this gut microbiome to treat health and disease phenotypes. And I know I'm short on time, but I just want to briefly say that in addition to doing research, I'm also an educator. After you finish your grad school and you go into doing a postdoc, there's a lot of different avenues for you to pursue. It could be going into industry, it could be going into academia, it could be going into science writing. There's all different types of ways you can take your PhD. And in my case, I want to go into a faculty position where I get to continue doing amazing research, but also teaching. And so one of the programs that I'm a part of is called Uplift. It's an NIH-funded transition award for postdocs to be able to teach. And part of my teaching responsibilities happens at a sister institution at California State University, Los Angeles, or CSULA. And these are some of my mentors as well as my colleagues, and we spend a lot of time driving back and forth in LA traffic. Well, we did, not so much anymore, but to teach at CSULA as well as do research. And so teaching is a great passion of mine. And if teaching is something that you find interesting, then doing academia is a great way to do it. Or you could be teaching without research. There's also that too. And so I just also wanted to thank everyone that's in my lab, my mentor, obviously, Elaine, all of the PhD, postdoctoral, and of course, all the undergraduates that have been instrumental in making this research project possible. And so with that said, I will take any questions.
We're a little short on time, so if you guys have any questions, just ask as soon as you can.
Sorry, let me see if I can read. How long did the research take? Oh, the research took many years. Different ones, different projects take much longer. And so at least for the maternal microbiome one, that one took around 4 years and a year in review in the journal before it got accepted. And yeah, it varies. It depends on how big your question is and how thorough you want to ask it. So I would say in most parts it takes years. And then do you take high school student researchers? We have taken high school student researchers. It's some of them came through programs, some of them came through recommendations. If you're interested in doing research, I would definitely look through the different websites, find a researcher that you're interested in, and just I would email them. I wouldn't cold show up in their office, but definitely go email them and take the initiative to reach out and see if you can get a research opportunity. I think we are, at least for most of us, having undergraduates or high schoolers is a great way to get started on research, especially during the summertime too.
Any last questions? Anybody? No last thoughts?
Yeah, and sorry, I didn't put up my email, but if anyone has any other further questions, you can email me. I don't know, I guess I could reach out to Diana and then she can forward that information to everyone.
Yes, definitely. I can send it through the Slack.
Okay.
Okay, awesome.
Thank you, everyone.
Yeah, thank you so much, Dr. Wong. I know it's very early and you're very busy, so thank you so much. I hope everyone really enjoyed learning about Dr. Wong's journey through microbiome research. And now we're going to transition to Dr. Olga Zaksybayeva, who is an associate professor of biological sciences and an adjunct associate professor of computer science at Dartmouth University. Dr. Zaksybayeva's research aims to understand how microbes change over time, and she'll be discussing the genomic clues to the origin, evolution, and diversity of life. So without further ado, everyone, please welcome Dr. Zaksybayeva.
Hi, hi everyone. First of all, thank you for inviting me. Okay, now I can share the screen. I was not allowed to share screen before. I'm really impressed with Diana's efforts to make this event happen. Do you guys hear me okay? Diana, can you?
Yeah,
okay.
Sounds good.
All right, sounds good. And then you guys see my slides, is that correct?
Yeah, we can see them.
Okay, great. Just, making sure that we are good on technical side. All right. And the other thing that I've done, depending on how your stuff is set up, you can, whoops, I don't know what happened there. You can put my video into the upper right corner here. I tried on my slides to leave that space completely open. So what I decided I'm going to talk to you today, I know my title is a little generic, is about, in general, how I got to do what I'm doing now, and highlight just one project that is going on that we're really excited about and been producing some interesting results. So just to go, let me also change. Sorry, there's a bit of a setup here. Just to go back to my high school time, sort of I have really fond memories of my high school, which was a special school. Was kind of geared towards physics and math education, but of course, you know, we had all other subjects, and I was really undecided what I wanted to do in the future. Had no idea. I actually didn't like biology that much. The way it was taught to us, it just didn't. I didn't click to me. I really like nature, but I didn't like it as a what I wanted to study. But it was interesting. I really like coding. So computer science, at that time wasn't called that way, was really interesting to me. I like chemistry, I like physics, math. I like geography a lot. I like geology or sciences, but I really wasn't undecided. And so I was good at math at the time. So I just said, all right, well, I'll go to college to study applied mathematics this way. I can also do some coding, and we'll see how where I get there. So how did I end up being a professor in biological sciences? Well, turns out when I was in college, we had an optional seminar where a professor, he was in the physics department, came to talk to us about life in the universe. And he was talking to us about how vast the universe is. And, you know, just to illustrate, I'm going to show you a picture that was taken by Voyager 1, which is the probe that was launched back in 1977. And when it was about 4 billion miles away from Earth, it sent back this picture. And most of the picture that you see here, this color pixels, are just noise from the camera. The only signal here is this little dot. And this pale blue dot is Earth. Everything else is pretty much empty space. And so in that lecture series, we spent a lot of times talking about how did life originate on Earth and how widespread is it in the universe? Kind of unknown questions, still unknown questions. And this is what was one of my turning points where I said, oh, this is so exciting. I should really do this. And moreover, the professor told us, well, you know, you guys are math majors. You can work in this from mathematical computational perspective, because to really understand life, we need to understand information that creates life. And every living cell has information. Whoops. And this information is encoded in our genome, which we can define as a complete set of genetic material that is present in every cell, including every cell in your body. And so here it is. This is just a small portion of human genome sequence. Lots of this DNA bases, T, C, A, and G, that we can computational just encode as letters, 4 letters, and somehow this stretch of information has instructions to create a human or any other living organism. So we're still trying to understand how we go from this code to a living organism, and understanding this will allow us to understand how life originated on Earth and elsewhere. And so this information is inherited. It's passed from one generation to another. You got your DNA from mother and father during this fusion of 2 gametes. Now bacteria pass information by duplicating the genome and then passing it to daughter cells during the cell division. And during this information transfer, there are DNA copying errors. And so it's actually not that frequent that they occur, but when they occur, they changes create these modifications that are descendant to the next generation, and that's what drives evolution, and that's what drives diversification of life as we know it. Now, what then we can do over time, we can use this accumulated errors or accumulated changes, now they're no longer errors, and we can go back in time. In a way, we can do archaeology of genomes. And I'm going to read this quote because I really like it, by Eric Lander. Evolution is, so to speak, an experimentalist that has been running experiments for 3.5 billion years since the origin of life on Earth. And wonderfully, the genomes of today's organisms retain the lab notes of these experiments so that we can go back and reconstruct the events that took place. And so just to illustrate it graphically, we have a present-day organisms. They have some genomes, they have this information. We can compare their genomes and we can track back the events that led to their diversification. I'm going to talk a little later about just kind of an overview of how we can do that. And hopefully, you know, we can go deep enough in time that we can, you know, peek at the early evolution of life, maybe to the origin of life, but at least to some early evolutionary events that took place. Now that's all really cool and exciting, and I was really, as an undergrad, was really excited about this. And then I had a chance when I was 18 to meet a Nobel Prize winner, Professor Manfred Eigen. who he got a Nobel Prize in chemistry, but he spent a pretty big chunk of his career studying the origin of life question. He said, well, Olga, yeah, it's really exciting. These are the important questions. But he said, you know, these are tough questions. And as a young scientist, you probably want to start small. And maybe you want to start, you know, identifying some smaller problems that kind of fit within the framework of understanding evolution, but maybe not directly address the origin of life question. And so the rest, as I say, is history. So I spent my graduate school years working in the molecular evolution lab. I learned a lot about biology, about molecules. I did too a lot of coding, bioinformatic work, and eventually ended up having my own lab, affectionately known as the Lab of Oz. And so what we really do in my lab, from sort of a broader perspective, is to understand the rules that govern microbial evolution. Again, with the goal that understanding the rules will help us understand how life originated on Earth. And so why microbial? Well, let's zoom in on that pale blue dot that is our planet. Although majority of life that we can see with our eyes is macroscopic, microbes actually rule the world. There's 100 million times as many bacteria in the oceans as there are stars in the known universe. And you just heard from Helen in a previous talk that, you know, even microbes within us constitute a high proportion of our body weight. And so it's not only dominated, the Earth is not only dominated by microbes now, it was dominated by microbes back and at the time of early Earth history. And so most likely when life originated, it was microbial. So that's why we're focusing on microbial evolution. But of course, you know, they're also very cool to think about. Now, going back to change via mutation that I introduced you earlier, this is not the only way microbes evolve. The microbes figured it out that this is not the most efficient way to gain some new function. Instead, what they do, they share the genes that are already known to do something. And that's called horizontal gene transfer. And to do that, they have all sorts of mechanisms. They can connect to each other using a variety of bridges that they create. And so through these channels, they literally can pass genetic information. So this is in a way similar to eukaryotic sex, but not mechanistically, just conceptually. But also, this gene exchange can occur via viruses. And so in the ocean, there are a lot of viral infections. You know, viruses are on our minds these days anyway, but think about this, that 20 to 40% of all bacteria in the ocean every day die from viral infections. And a lot of viruses known to carry genes that are not really viral genes. For example, just a couple of headline titles of the papers. For example, they can carry photosynthesis genes. This would be the genes that bacteria that can do photosynthesis have, that allow them to harvest the sunlight. But a lot of viruses carry those genes that they can prolong the life of the host, so it can photosynthesize. The same idea that phages or viruses, that's just bacterial virus lingo, bacteriophages, can spread antibiotic resistant genes because they carry them in their genomes and they infect different bacteria, including those in our gut. All right, so now I'm going to just focus on one project that is about a very strange bacterial virus. We're very excited about this project. And let me introduce you what this strange virus is. We're going to think about a specific bacterium called Rhodobacter capsulatus. It's an aquatic bacterium, lives in lakes. Its relatives live in oceans. And so something funny happens when these bacteria are crowded together. About 0.1 or 3% of the cells in this crowded population start to undergo transformation. They turn on about 24 genes that previously were turned off. And when they turn on these genes, they start to produce what looks like viral particles. So this is an electron micrograph of what they produce. They're very small nanoscale particles. So it looks like viruses, virus-sized. And then, unlike viruses usually package their own DNA so they can propagate and replicate in another bacterium. These guys don't. They carry a random piece of Rhodobacter capsulatus, the bacterial DNA. And then they kill the bacterium that produces them. And then these particles get released to the environment. And what they can do, they can deliver this DNA to other cells by infecting them, but there's no viral genome in there. So the question is, what is this? They were dubbed gene transfer agents because they're not really viruses, but it looks like they can transfer genes, so GTAs for short. And so in my lab, we're studying what are these GTAs, what are they for, and how did they evolve? And so what are they for? Well, they could be for a variety of functions. And so here, this is just a cartoon illustration of hypotheses. For example, they can help other members of the population to provide genes that fix DNA damage that could be caused by UV light, or maybe, they find the population of this bacteria find themselves in an environment where only some of them have some genes, others don't, so they can obtain new useful traits. By the way, this is a side project of my undergraduate student to make a children's book about this project, and we're in the process of trying to find a publisher. So I'm using some of her artwork here, Emma Esterman, who just graduated. So these are just hypotheses. They need to be tested, but these are possibilities. And so what we do know that these GTAs evolved from what was regular virus in the past some long time ago. So the question is, when did this happen and how did this happen? And so this is where we can use our archaeological techniques that I introduced to you earlier to try to figure this out. And so let me step back and talk a little bit about conceptually how we do that. Let's imagine you have an ancestral DNA sequence, and over time it starts to accumulate changes. So the changes here are shown in yellow, and it starts also to diversify. So you have now one variant that got these two changes. You get another variant that has these two changes. And now let's imagine that these are our contemporary bacteria, and this is their ancestor. Now, when we sample data now, this is a black box. We don't know what's happened, but we only have these 2 pieces of information, but we can model how these genetic changes came about. And there are a lot of models available, and I'm not going to go into details of them, but the idea is that using these models, we can reconstruct events that gave rise to these sequences. And then we can see what happens. All right, so again, this then would lead to this tree of past events connecting the contemporary genomes. That's just a picture that I showed you earlier. But now what you look here is the actual data. Ignore the labels, it's just different names of related bacteria. The Rhodobacter capsulatus is within this wedge here. And so what we've done, we looked at DNA sequences similar to those of Rhodobacter capsulatus and asked the question where the GTAs are. And we find that everything shown here in green, there's this big group of related organisms that actually have GTAs. And it looks like, just based on inference, if they all have it, the ancestor must have had it. So somewhere here, we think it originated somewhere here in the past or earlier maybe. And the other technique that, again, I cannot today go into details, but we can use fossils and other evidence to actually date when this happened. It's not always very accurate, but we can put some kind of a range, which is, you can see here, between 800 and 1800 million years ago. So pretty wide interval, but quite ancient if you just even look at 800 million years ago. So this GTA has been around for a while. And so this is not just some kind of a new defective virus. It's maintained in a pretty large group of organisms, so bacteria. And what we know from evolution of microbes is that if they were not using this, they would have gotten rid of it. It's expensive to maintain useless DNA in microbes. They try to get rid of things. They're very minimalistic. And so we think these GTAs are actually maintained in these genomes. And so the next thing that we've done, we actually looked at amino acid compositions of proteins that make up GTAs and compare them to viruses. And what we found that they're actually very different. So what I'm showing you here is just an example of one gene, doesn't matter what it is, but we just basically counted how many of the 20 amino acids that are known to be used by any organism on Earth are present in this viral protein versus GTA protein. And we just color code them here. So you can just by eyeballing see that the color bars for GTAs versus viruses are different. And I'm not going to show you here the data, but we actually demonstrated that this difference is statistically significant. So they're not just some kind of a viruses that somehow just weird ones. So there is a difference, and so this difference is so pronounced that we can actually use a machine learning techniques to train our models, machine learning models, to distinguish GTAs from viruses just by looking at the amino acid composition. So we developed a program in the lab called GTA Hunter. And so basically this program now, if you give it a gene that maybe GTA, maybe viral, it will spit out the answer, what it thinks it is. And by applying this program to many bacterial genomes, we actually found a lot of sequences that potentially are GTA. So we think GTAs as a phenomenon is much more widespread than we think it was. All right. So now this change in amino acid composition is also not random. It turned out that this amino acids that GTAs use is skewed towards energetically cheap amino acids. And here again, I'm showing you a piece of the data for the same gene that I showed you before. And the way we quantify what energetically cheaper means is how many carbons are in amino acids. So the more carbons there is, the more expensive it is to produce an amino acid. And if you have more amino acids with larger number of carbons in a protein, it's more expensive to make that protein. And so what you see here is how many carbons per amino acid side chain is present in GTA on average versus in viruses. And you can see here the viruses have many more than GTAs, and it's actually statistically significant, and we can show it that it's true for other genes as well. So this analysis then allows us to create a working hypothesis that GTAs are devices for survival under energy or nutrient limited conditions. Maybe this is one you want to have some new genes or have DNA repair happens. Now this hypothesis will need to be tested in the lab. In the actual experimental lab, we do computational predictions in my lab. So I'm trying to convince some collaborators to do some experiments. So, so that is cool. We could take some computational analysis and make predictions that could be tested. Now there is another puzzle that is completely unresolved about this system, is how this system is maintained over time. Because if you remember what I told you earlier, the bacterium that actually produces GTAs get basically blown to release all those GTAs. So the question, if it gets killed, how do these genes actually survive in a population? What is a driving selective factor? It's a fascinating question to which I don't have an answer for right now, but we're working on modeling and doing some analysis for this. All right, so you know, this, a lot of people tell me this is very esoteric, and why would you study this? And is there anything about GTAs that could be useful for practical and societal application? And the answer is yes, there is. There is actually a company that is interested in looking into this system. It's far from being implemented, but we, in general, could use virus-like particle as a gene therapy, and it actually has been used as such. And so understanding in general how the genetic exchange occurs in something that looks like a virus, whether it's in bacteria or not, can advance our gene therapy efforts. But you can also think about treating of bacterial infections with engineered bacterial viruses. We right now running out of antibiotic treatments, and so maybe targeting and first of all designing bacterial viruses that carry basically something that can kill infectious bacteria could be achieved via GTA-like particles. So just to summarize what I hope I conveyed in my talk. So I study computational biology. I like it because it, first of all, allows time travel and can also provide insights into biological puzzles that you can't necessarily get to by experiments, but I collaborate with a lot of experimental biologists to test our hypotheses. And in general, studying microbial evolution can shed light into rules of life, to more of a basic question about how life evolves. And then finally, more specifically to this project, acquisition and modification of already existing genes is an important evolutionary force. Microbes maintain many mechanisms for such gene exchange, and GTAs may be one of them. Now, in the final couple of minutes, I just wanted to say that a lot of the work that I was presented was not done just by me, but by members of my lab. That included postdocs, graduate students, and a lot of undergraduate students. And what you see here in parentheses is where those students are. If they don't have anything in parentheses, they're currently in my lab. Otherwise, you know, they work in different places. A lot of undergraduate students went on and into PhD programs. Specifically, Danielle and Taylor and Camille worked on that GTA Hunter program. They actually designed it from scratch. They over years we worked on this. Emma Esterman She did not just book, but analyzed a lot of genes from GTAs. And currently, Jonathan and Jack continue to work on this GTA project. Now, the other thing that I want to mention, that in my lab, I have both computer science and biology majors working. Depending on the project, they either focus on the coding part or more biological interpretation part, but they get to interact with each other and you know, learn both disciplines. And here's just thank you for a lot of funding. So with that, I can entertain some questions if I figure out how to get out of this. I think I can stop sharing. And thank you for your attention. All right, I think if you put things in the chat, I can read them. I think we're okay on time, right? Sort of.
Yeah, we have about 5 more minutes. To anyone watching the live stream, I know there's a bit of a lag, but we're also moderating the live stream. So if you have any questions, you can pop it in the YouTube chat as well. Everyone's very non-inquisitive this morning.
I have one question actually. Since I'm a CS person, how can I, how can I get involved in like research like yours once I'm in college, right? What do you recommend for CS majors to be doing?
Yeah, that's a great question. Actually, you know, a lot of times I just get inquiries. So I think as Helen was saying in her previous talk, a lot of times it's just you need to be proactive, and you need to find a person whose research is of interest to you, and contact them. And usually I talk to a lot of students, and if I have openings, my lab is pretty small, but you know, students graduate, and so if I have openings, I usually chat with the students and see what kind of common interests we have, where do they want to get with this project, and we discuss possible projects. It's usually a kind of, it's not me telling you, okay, if you want to join my lab, you're going to do X. It's more like, hey, what are you interested in? What am I interested in? Let's find, you know, what can we work together on? So that's kind of an advice. Just find a professor who does something that is of interest to you and reach out. And you know, if one professor says no, you know, there are multiple reasons why that could be. It's not about you. It could be maybe, you know, they already have 5 other students. So don't be discouraged, and keep asking several professors. All right, I think there are some questions. I'm going to start with the first one. When were GTAs first discovered? Oh, that's a great question. They were actually discovered back in 70s, I think 1975. Nobody paid a lot of attention to them. They were kind of obscure for a while because people thought like, oh, this is weird. Okay. And I think a lot of, at that time, there was just not a lot of genomic data. Well, none of the genomic data was available at that time. And I think we are right now in biology in this unprecedented time when we can just look at the genomes and infer a lot of stuff that was not available. It was just harder to study. So right now GTAs are getting more traction because we can see them without actually seeing them in the cultures. Do you take a high school student? And the answer is yes, it depends. A lot of times, again, it's by contact. We do have programs here for high school students. So most of the time, if I will take a student, I will take it through the program, just because this way it's the students will get additional experience, not just, you know, working in my lab, but also getting other professional development advice. Would you need any lab experience before joining a lab as a curious high schooler? Well, I usually, I get a lot of inquiries, and of course, I cannot take all of the students. I usually end up with taking 1 or 2 summer students, for example. If you know how to code, it's a plus, because a lot of time, everything pretty much what we do in the lab requires, like, for example, Python coding. And it's hard for a very short, say, summertime for you to start from scratch and just not knowing anything. So a lot of times I would definitely give a preference to a student who already knows how to code, then they can come and work on the project a little bit, more meaningfully. Question from YouTube live stream. What are some COVID-proof research project ideas for high school students? Anything that doesn't need a lab or going to a public place? Well, so I haven't been, right now I'm not at Dartmouth, and I'm in my house in rural Vermont. All of the work that we've been doing, including what I presented today, have been done remotely. My graduate students and my undergraduate students are working remotely. We have computer servers to which we connect, so all the work is done on those servers. We communicate via Zoom. We haven't been to the lab. We have all our meetings, group meetings over Zoom. We work like that. We use Slack as well to communicate. So yes, a lot of computational work is actually done virtually, and my genomics class this fall will be done virtually as well. All right, looks like this is all for the questions. All right, well, thank you everyone for tuning in.
All right, thank you so much, Dr. Zaksibayeva. I hope everyone really enjoyed her talk and learning about GTAs and bioinformatics and computational biology. So next we have Dr. Isaac Sandoval, who is an assistant professor in biology at Dartmouth University. Dr. Sandoval received his MS and PhD in chemical and biological sciences at the National Polytechnic Institute in Mexico City. And just a note, I earlier said Dartmouth, and to say Georgetown University. He focused on physiopathology and host immune response during his graduate work, and has now moved into a more challenging field, neuroscience. So Dr. Sandoval will be discussing the science behind memory forgetting. And without further ado, please welcome Dr. Sandoval.
Hi, everybody. Can you hear me well? Can you see my screen?
Yep.
Okay, thank you. Well, first, I want to thank Diana for this wonderful invitation. I think I always have thought that it's very important to communicate science to young guys, to communicate the excitement that science produces. Ever since I was a kid, I knew I wanted to be a scientist. I don't know how that happened in me, but I never had doubts about what I wanted to do. And in biology in particular, I was always very interested in knowing how live things work. I remember when I was a kid spending a lot of time looking at animals or insects and just wonder what was going inside their bodies to behave like they behave. For me, they were like small machines, and it was just so mysterious to know like what's going on inside their bodies that makes them alive. So becoming a scientist was not a question for me. And then I just want to convey to tell you that there's nothing more enjoyable than finding something that nobody knows. Like when you're in science and you were conducting an experiment and all of a sudden you have an epiphany or you find something that is completely new and nobody knows about it. It's just the amount of energy that you get from that is just amazing. It's one of the best experiences you can have. And so that's why I became a scientist. And I started my career, I am from Mexico, and I started my career working on a parasite that infects the brain, especially of kids. And it's a very dangerous disease. But then I remember when I was in college around the year 2000, I was reading about this this gastropod in the ocean called Oplecia. And I was reading that a guy got a Nobel Prize because he discovered how, or the molecules that are involved in how the memories are encoded in the brain. His name was, Eric Campbell. This is his picture. And this is the Oplecia. This is a mollusk that lives in the ocean. And using this animal, he started to discover how the memories are encoded in the brain. And I was in college when I read about him, and it was just fascinating for me. And so that's why I decided to go into the field of neuroscience, and particularly in learning and memory, to try to understand how our memories are encoded in our brains. I have to say that I got to meet Dr. Kandel a few years ago when he visited the institute where I was working in Florida. And so this is some of the things that are also very exciting when you get to know the people that inspire you to in your career. And he was one of my heroes. I have many, but this guy was the one that made me decide to go into learning and memory field. And so my lab now is trying to understand exactly that, how our memories are encoded in the brain, how the memories are consolidated, meaning that how these memories that at the beginning, when you learn something, this memory is very labile, it can be forgotten very easily. But some memories, if they are really relevant to your life, then they become what we know as long-term memories, memories that can last for a very, very long time. And that process is called memory consolidation. So my lab started to work on how memories are encoded, how memories are consolidated. But very recently, we are also very interested in trying to understand why do we forget and how do we forget? What are the molecular mechanisms behind that memory forgetting? And so usually when we think about forgetting, we think that forgetting is something bad, right? Like, because we associate forgetting with some very devastating diseases like Alzheimer or dementia, and also forgetting something important can get you into real trouble. I remember one time when I was in college, actually I was in my PhD, and they invited me to give a talk on whatever my research I was doing at that time in a city that it was far from Mexico City. So this city was like 4 hours far from Mexico City. And I remember this talk was in a very important museum, and it was a big deal. And so I decided to use a nice jacket and a shirt and a tie. So I remember I traveled the day before my talk, and so I packed everything in my suitcase. But I decided to put my jacket, my shirt, and my tie in a different bag, a bag special for that. And so I traveled the day before, and this talk was in the morning. And I remember I woke up with enough time, I took a shower, and then when I was about to dress up, I realized that I forgot my jacket and my t-shirt and everything, my shirt. And so that was very scary because I didn't have any other clothes proper for this talk. So I went out in the city and tried to look for a store where I could buy something, but everything was closed. It was really, really early. So I was just like knocking in doors of the stores, but hoping that somebody was inside them. And yes, I found somebody that they were about to open the store. So they opened it for me and I got to buy something, and it didn't fit well, but it was enough. So yeah, I mean, forgetting is if you forget something important can get you into real trouble. And I'm a very forgetful person. This has happened to me many, many times. And so when many times I found myself thinking, it would be wonderful if I don't forget anything, if I could have a perfect memory. But then when you think deeper about this, you start to realize that forgetting maybe is not such a bad idea. And this is very well represented by a fictional character called Funes. And this is a drawing of him. This is a fictional character of a book from the Argentinian writer Jorge Luis Borges. And this guy was a guy that he could not forget anything. For him, his memories were perfect representations of the past, something like a videotape. And so he could remember all the details about everything that he experienced. And And that was okay. I mean, when somebody told you that, it sounds like could be fun. I can't remember everything. But he also had a bunch of cognitive problems. Like for example, when he was trying to remember one day of his life, it would just take him one full day to remember a day, right? Because he remembered every specific detail. And so he could not live in the present. He was constantly living in the past, because also every time he saw something that triggered memories in his mind, he was just going back to the past, and he was not able to live in the present, along with many other problems. And so when we think about, intuitively, we think about what forgetting is doing for us. Why is it important that we forget? We came out with at least 4 possible causes. So one is that if you think that your brain has a limited capacity to store information, like the memory of your computer, and you experience all these things every day, the colors in the sky, the people walking, the things you read, the things that you see on TV, all this information, if it will be stored in your brain, it's so much information that probably it's going to saturate your brain pretty fast. And it will leave you very little free space. But that doesn't seem to be the case. We always have more capacity to learn new things. And we think that forgetting is really important for that. So getting rid of things that are not important anymore, that you don't need, and then you just can just get rid of them. And so it's important for that, so to be able to have free space in your brain. Another cause is we think that forgetting is really important to increase efficiency during memory recall. And I'd like to give this example. Let's say that this desktop of this computer is your memory system. And all these files here are the things that you remember. Now, let's say for a moment that you need a file from all this mess, and you need a picture or something from this desktop. And then if you try to look through all this mess, it's going to take you some time. And it's going to be very slow to find the piece of information that you need from all this mess. On the other hand, if you keep your system clean by means of forgetting, so again, getting rid of all the information that you don't need anymore, then your system is going to look more like this. And it's going to be way easier and faster to find what is important for you at the moment. And so forgetting is really important for this efficiency during memory recall. Another thing is that we live in a very changing environment, right? So our memory, our environment is changing all the time. Things are changing all the time. And so forgetting is really important to maintain what we call behavioral flexibility. So you need to be able to update your memory system to forget things that are not important anymore and to update it to new situations. For example, let's say you live here and you have a favorite restaurant and you know exactly where it is, you know exactly what path to take to get there. But let's say they move this restaurant, and then now you need to update your memory system. Now you need to update the new location and to learn where is the new location of that restaurant. And with time, you're gonna forget about the old location. And so memory forgetting is important for this kind of updating, memory updating. And lastly, forgetting is really important for promote generalization. And I'm going to explain you what this is. Let's imagine that you are a mouse or a rat or something like that, and then you have encountered several cats in your life, and now you know they're dangerous because they chase you, they try to eat you. And let's say these different cats have different colors, different sizes, different size of fur, different odors. And if your memory system was a perfect representation of each one of these cats, then your memory system will look like this line, okay? So let's say these blue dots are each one of these cats, and their position in this graph is given by their different characteristics. And so if your memory is perfect, then your memory is going to look, your memory to predict what a cat is going to look like this black line, okay? Now let's say you find a different feline, like this is a bobcat, okay, which is completely, not completely, but somehow different. It's bigger, it has different scents, different color, and that's represented by this green dot here. Now, if your memory is perfect, then this memory is going to fail to predict that this is also a feline and this is also dangerous. And so this can be very bad for you. On the other hand, if by means of generalization, you forget about all the small details of every specific, but you just keep the gist of the information of what a cat is, what a feline is, then your model to predict what a feline is going to look more like this black line. And then this model is going to be better to predict that this bobcat is also a danger for you. And so this process is called memory generalization, and forgetting is really important to achieve this. Okay, so in my lab, we are trying to understand how do we forget? What are the genes or the proteins that are involved in this process of memory forgetting? And to do that, we use the fruit fly. This is the animal model that we use. This is called Drosophila melanogaster. And we use this animal because it has a lot of advantages. So it's really small. It has a great amount of genetic tools. People have used this animal for over 100 years. So they have developed a lot of tools that allow us to uh, dissect their brain in different ways, to mess up with their brains, to control their brains. And so it's a very, very useful model. Also because it's so small and they can live in with very little, it's a very cheap model to work. It's low cost. And also in time, we can have as many flies as we want in around 10 days. That's the time of the life cycle of a fly, compared to, for example, animal models like mammals, like mouse or rats, where you have to wait for months to get animals and start working with them. And you can have as many, and they're expensive. So Drosophila is a great model for that. So how we do this? Well, the first thing we do is we train the flies. Okay, so we teach them stuff, and the fly can learn. What we do is we put small flies in a classroom, and when we lecture them about the dangers of electricity, and they learn. I hope somebody's laughing because that was a joke, but we do something similar. Okay, so what we do is we put flies in a tube, an empty tube that looks like this. We put a bunch of flies there, and then we could pass an odorant through that tube, and so they can perceive that odorant. And then at the same time, we give them like mild shocks to their legs. And so normally the protocol works like this. So we put the flies at the presence of another one for around 1 minute. And then at the same time, we give them 12 shocks of 90 volts in their legs. And so they associate the odor with something bad, with the danger. And they learn this association. We then let them rest for 30 seconds, and then we present a second odor for 1 more minute. And this odor is not paired with anything. So it's just odor. After this learning session, we can put the flies in a teammates that look like this, and we can present other one and other two in two different arms. And then if the flies remember that association between the odor and the electric shock, they are going to avoid it. So they are going to run away from there and and they are going to position themselves to the other arm with the other odor. And so in this way, we can calculate the memory. We can estimate how much they remember from that task. This is one of the things that we do. So we do behavior. We analyze the behavior in this way, how much they remember. But we can also look into their brain. So we have these powerful microscopes where we can put a fly under the microscope, and then using some genetic tools and some fluorescent proteins, we can actually record the brain activity of a fly. And so what you see here is a fly trying to walk around. It's fixed in a chamber that is below this microscope, and then it's moving, it's alive, it's trying to walk, trying to fly. And at the same time, we are recording this video that you can see here in the upper panel is the brain activity. So every time you see these flashes of color, that means that the brain is very active. In this case, we are recording from this area of the brain, which I'll present you a little later. And so in this way, we can also train the fly under the microscope. We can put an electric reed touching their legs, and we can deliver others. And so we can train the fly while the fly is learning, and then look at the brain and see what is going on in the brain as they learn or as they forget. We do also a lot of microscopy, so we can look with a super resolution microscopy. They're part of the brain that is important for memory encoding. In this case, we are looking at this region here called V1, and this is a lot of detail of all the synaptic connections, so connection between different neurons and how they change with learning and how they change with forgetting too. This is the Drosophila head, and this is their brain. This is how it looks. These two lobes that you see here, they're the optical lobes. They're the ones that receive the information from the light. And then these green things that you see here, they're called olfactory bulbs. And they receive the olfactory information. So their fly noses this thing that you see here. This is the antenna. And so others are perceived with this antenna. And that information is sent to the brain, to this area. And that information then is conveyed to the back of the brain and gets to these L-shaped structures here called Martian bodies. And this is exactly where the memories are encoded. These are the sites of the brain where the memories get stored. And also, of course, is the place of the brain where the memories are forgotten. Okay, so this is a kind of a complicated model of how we think memories are encoded in the brain. Normally, the cells or the neurons that perceive the others are called Kenyan cells. So these are represented by these green circles here. And then once they receive the information, that information is going to go down and is going to be conveyed to two different kind of neurons, these blue ones here and these pink ones here. And these neurons are going to drive the approach behavior. So when you activate these neurons, the flies like it, and so they will approach to whatever stimuli that they are sensing is rewarding. And on the other hand, we have another branch, another kind of neurons that when these neurons are activated, they avoid whatever is causing this activation. They don't like it. So that is an aversive stimuli. And so normally when we have another that is neutral, that it doesn't cause any behavior, the flies don't care if they don't know if it's good or it's bad. One of these cells is going to be activated, and then it's going to activate these two branches in a very equilibrated way. So at the end, the result is nothing because you have the same amount of approach, the same amount of avoidance, and so the result is no behavior, nothing happens. Now, during aversive learning, what happens is that you have the activation here of a specific odor you're using for training the fly. And then at the same time, you have the aversive stimuli, right? So here you have the electric shock that is conveyed by these neurons called dopaminergic neurons. So these are called dopamine neurons that produce dopamine. And then when you have both the stimuli presented at the same time, you have a point in this circuit where there is a coincidence. And that point is going to be depressed, basically. And so now what is happening is that the next time that the fly experiences that same odor, that information is going to go down again. But since this node or this point is now depressed, that is going to result in less activation of this branch. And then the result is a reduced approach. And since the other arm of this pathway is still normal, and then now the avoidance has a higher weight, right? So the balance between a reduced approach and a normal avoidance is avoidance. And so now after aversive training, the fly is going to avoid that particular odor. And so that's what's happening in the brain. And the same thing or a similar thing happens with appetitive training. So we can also train the flies to associate odors with a reward. In this case, we feed them with sugar, and very similar, something happened in that case. Now the avoidance branch of the circuit is reduced and the approach is still normal. And so the end result is the flies like this odor and now they approach it. Now, when we train the flies, we can score that memory and we call that performance index. So what this means is if we have a score of 1, that means that all the flies remember the task. So all the flies avoided the odor that was paired with the shock. And if the score is 0, that means that they don't remember nothing. And so when we train the flies and we test their memory right away, most of the flies remember the task really well. But if you test the memory like sometime after training, they start to forget, just like we do. And if you test the memory around 24 hours later, most of the flies forgot the task by that time. Now, just like in humans, I know you experience this, right? So when you, let's say you have a test and you study once, only once, and you have this test right away, maybe you can do okay with one test, one training session or one study session. But if you wait long enough, at some point you're not going to remember anything. But let's say instead of studying only once, you study many times, and you have you study, let's say now, and then you have a rest, and then you study again, and then again and again. If you study many times, then this memory is going to last for a long time. And the same thing happens with flies. If we train the flies 5 times, they will remember for a long time. This memory can last many days, even like 20 days or 30 days. And taking in consideration that the life of a fly is around 60 or 50 to 60 days, that means that this memory can last half of the time of their life. It's a very long-term memory. And this kind of memory requires protein synthesis. So that means that new proteins have to be synthesized in order to form this long-lasting memory. Now, so when we wanted to understand what are the genes or what are the molecules that are involved in this forgetting process, we reasoned, okay, let's do something. We can manipulate genes in flies very easily. And so what we do is like, okay, let's mess up with the function of a gene and see how that affects their memory, and see if that affects how they retain their memory. If maybe we can have a fly that remember for a long time. And so we did that. We messed up with the function of around 3,000 genes. That took around 3 years and the work of 4 or 5 people. And at the end, we found a series of genes that look like this. And this is one of them. This is called Scribble. This is the name of the gene. And this is a 3-hour memory score. So that means this point here, when we train the fly, we wait 3 hours and test the memory. And these are control flies when we don't do anything with the gene. And this is when we interfere with the function of the gene. Okay? And so you can see how these flies actually remember better. Now, this gene was discovered in the year 2000 and by a professor called Builder. And he found that this gene was expressed in the epithelial cells, basically. So in the cells of your skin, in the cells of your gut. And this protein was really important for what is called cell polarity. So if you take cells from your gut or cells from your skin, and you will notice that the top part or the apical part, the top part of your cells is very, very different from the bottom part of your cells. And Scribble, this protein called Scribble, is really important to make that difference. And that difference is fundamental for the function of your cells. So if you don't have this, then you cannot survive. And Scribble is really important for that. And then we found that this gene, it was also expressed in the brain, and it was important for memory forgetting. So when we knock down that gene, or when we interfere with the function of this gene, this is what we see. This is the memory curve that that we found. So no, this black line is the memory decay of a control flies, or of control flies, and this is the memory decay of flies where we basically interfere with the function of this gene in the brain. And you can see how these flies remember way better. Do we still have time? Sorry. Yeah, Diana, how much time do we have? Sorry.
Yeah, we have, right now we're approaching the 30-minute mark, so we have a few more minutes.
Okay, sorry, I'm going very slow, but yes. So this is what we found. Messing up with this gene makes the fly to remember better. And then if we look and look at the memory of these flies for a very long time, these flies remember this memory for almost 60 days later. So they basically never forget about this. These are super smart flies. So we reasoned that there could be at least 3 reasons why this is happening. One, maybe the flies just learn better. They maybe they are super smart. They study one time, or we teach them one time, and that's enough for them to form a super robust memory. So memory acquisition. So they are just better at acquiring the memory. It could be that they have enhanced consolidation. So that means they are better at taking a label memory and transforming that memory into a long-term memory, a protein synthesis dependent long-term memory. Or it could be that these flies are impaired in memory forgetting. And so we did several experiments to try to see which one of these three options is what we are seeing in these flies. And the first thing we did is that we trained the flies with a mild training protocol to see if these flies were learning better. Okay, so that happens to you too. If you study just a little bit, you won't learn as well as if you study a lot, right? So here we train the flies instead of using the 90 volts that we normally use. We use 45, 20, or 10 volts, and you can see here the control flies and the scribble flies in red, and you can see there's no difference. So there's very little difference. If any, it looks like the flies are worse to learn. And this is more evident when we change the number of shocks. We normally use 12 shocks, but when we use 1 or 2 shocks, it looks like actually these scribble flies are worse at the acquisition part, worse at learning. So no, it's not this. They don't have an enhanced memory acquisition. Now, we knew that protein synthesis is important for taking a label memory and making a long-term memory. And so we wonder if this is what it was happening with Scribble. And so what we did is we trained the flies, and then we treat them with a drug called cyclohexamide that basically what it does is blocks protein synthesis. So this drug don't allow the body of the fly to synthesize new proteins. And normally in control flies, that is going to cause a decrease in the long-term memories. But you can see here how the Scribble knockdown flies, there's nothing. Nothing happens with this drug. So that means these flies are not forming protein synthesis dependent long-term memory. It's just a normal memory that lasts a very, very long time, but it's different in nature. And so it's not memory acquisition. And so what it got left is, okay, let's say if they have an impaired forgetting. And what we did here is something very simple. So we trained the flies to associate the other A with the shock, and then we present the other B with no association. And then right after that, we say, hey, no, that was wrong. Now let's learn this new thing. Now the other B is paired with the shock, and the other A is not. So normally what happens in control flies is that this new learning phase is going to interfere and is going to induce a forgetting of the initial learning. And you can see this by this graph. This is the initial learning, and then after the interfering session, that memory is decreased. So that means the interfering session has induced some forgetting. But that doesn't happen in the Scribble flies. Scribble flies, they just don't forget. They remember forever. Okay, so conclusion, these flies have an impaired memory forgetting. Then we wonder where in the brain is this protein required? And so using all the nice genetic tools that we have in Drosophila, we can actually go and interfere with the function of this protein in cell-specific manner. So we can decide where in the brain or what neurons in the brain we want to interfere with this gene. And so we found that when we interfere with the function of this gene in the dopaminergic neurons or the motion body neurons that called Kenyan cells, we have this enhanced memory. And so Then we did some microscopy and we found that actually Scribble seems to be expressed here. And this is what you can see here in green. Okay, so this is the Drosophila brain, and the green part is basically a technique to detect that protein called Scribble. And you can see how it's highly expressed in these L-shaped structures called motion body, which we knew are the parts where the memory is encoded. And so these kind of experiments now allow us to understand better what are the genes and what are the molecules that are important for us to forget memories. And it's not a surprise that this gene was then, a few years later, found that it's important for memory processing in humans too, mammals and in humans. Not only that, we also found that sleeping is really important for memory retention. So in the fly, one of the other tools that we have is that we can install switch in neurons, either light switch or temperature switch, but basically we can install switch in neurons and then we can turn on or off neurons at will. And so we knew, but some other researchers found what neurons in the brain are important for sleep. And so we decided to install a switch like this in the sleep neurons and then turn it on. And basically what you do is you put the flies to sleep. And this is what you can see here. This is a measure of sleep. This is the control. These are the control flies. And then when we turn on that light, that switch, they basically sleep more. And that resulted in a better memory. Okay? So forgetting or memory, like the memories are protected from forgetting when the flies are sleeping. And that's similar, something very similar happens in humans. So this is our final model of how we think things are happening. We found not only Scribble, but we found many other proteins that are important for memory forgetting. And so we believe that really what is happening is that Scribble is somehow like a scaffold. So Scribble is a protein that is binding to many other proteins and is putting together a signaling complex that, that is conveying the forgetting signal to the canyons and to that neurons, those neurons that are important for memory acquisition. And so we now know all these proteins, and all of these proteins are involved somehow in modifying the skeleton of a cell called cytoskeleton, and that is what is inducing the memory forgetting. And so this is a work that took, it took from 2010 and up to today, and this is what we have found so far. Okay, I just want finally to thank everybody for listening. This is my, these are the members of my lab, some the technicians, undergrad students and grad students. And I also want to thank my collaborators. And you can visit our website at this link here, cervanteslab.org, and learn more about what we are doing. This is a very nice picture of Georgetown. And if you ever interested in how we forget or why we forget, we can work together. Thank you.
Thank you so much, Dr. Sandoval. We can take like maybe a few questions if you guys have any right now. Oh. Dr. Sandoval, can you see the chat? So
that's what I'm
one question. Okay.
Okay.
Okay, cool.
Can you read it? I don't see.
Yeah. Can we tamper with Scribble KD and human brains so our memory is enhanced for school exams?
Yes. Unfortunately, we cannot do that now. We cannot do experiments with humans. People don't like it. I don't know why. No, well, yeah, we cannot do that. But there's some efforts on trying to develop drugs that will interfere with these genes. So we cannot manipulate the genes like we do in animals. But we can develop drugs that interfere with the function of those genes. And so in a way, try to improve memories, right? For example, especially in some pathological conditions where memories are impaired. In normal humans, like in the normal cognitive functions, I don't know if it's a good idea to mess up with the forgetting part. It could result in some other problems. Like I say, forgetting is normal. Forgetting is something that happens normally. It's something that is important for us to function normally. So you don't want to mess up with that in a normal condition. Okay, I can see the chat now.
There's one. Are there studies being done to explain why we forget some memories
Yes.
more easily than others? Yeah.
Yes, this is a really interesting question, and this is something that we have asked ourselves in the lab. And if you remember that curve of the aversive memory, and when we train the flies with electric shock, they do forget pretty easy, and then more or less 24 hours later, they don't remember anything. But I didn't mention, but if we train the flies with the appetitive conditioning, that means something good with sugar instead of the punishment, they do remember that for a very long time, okay? So that memory lasts way longer. And we are trying to understand why is that? How come some memories got lost really easy and some last forever? And in this case, it seems like it is the taste of the sugar combined with the caloric content of the sugar that somehow our brain can detect and basically signals to say, okay, this is really important. Remember this because this is important for you, for your life. This is important for finding food. And so those kinds of memory get encoded in a different way and they last forever. And so there are many ways to approach this question, but basically, if the memory of what we are learning is really relevant for our life, then the chances that it forms a memory that will last longer are high. And we are trying to understand why. And probably it's going to be different for every case, but yes, one of the things that are really important in humans and in mammals is the emotional part of a memory. If you, let's say, I'm sure you can remember, or you cannot remember actually what you were having for lunch, let's say 2 years ago. It would be really weird if you can't remember. But if something else was happening at that time, like for example, there was an accident or there was like the 9/11, I mean, you're probably too young, but something was happening in the world that was really important and it caused a really strong emotion in you, then you can remember all the other things that happened around that time very well. So you can remember how you had for lunch that day. You can remember what you were wearing that day. And so it's really clear now that emotions, strong emotions are somehow enhance how we remember things. Let me see other question here.
I don't want to cut anyone off, but it is 10:52. We're a little bit behind schedule, so maybe
Okay.
just wrap up one more thing.
Okay, thank you everybody for listening.
Yeah. Okay. Thank you so much, Dr. Sandoval, for joining us. I hope everyone really enjoyed learning about the science behind, encoding and the consolidation of our memory. So now we have Dr. Xia Yang. She's a professor of integrative biology at UCLA, and she's joining us to discuss how she dissects complex human diseases through big data bioinformatics and systems biology. Dr. Yang is currently a tenured professor in the Department of Integrative Biology and Physiology and a faculty member of the Institute for Quantitative and Computational Biosciences at UCLA. So Dr. Yang, please take it away.
Thank you so much, Diana. I really appreciate the opportunity to share our science here. And this is a really very enjoyable event. I'm listening and seeing the very interesting questions and learning new science. It's a really awesome opportunity. Thanks again for inviting me here. So yes, I'm going to share some of our work on big data informatics and system biology and using that to understand the complex human diseases. Okay. So I'll first go through the multidimensional complexity of human diseases so that, that build the foundation on why we even need to use this system biology approach to understand the complexity. And I then introduce some, briefly some work we have done in this area to learn about the complex diseases using the approaches. And lastly, I just wanted to highlight the importance of bioinformatics and big data analytics in resolving complex diseases. Okay, so first of all, let's go through some of the complex diseases we're talking about. Many of these complex diseases I'm showing here are the leading causes of death, like heart disease, cancer, stroke, Alzheimer's disease, diabetes, etc. Right? So this is the data in the US. In the world, some of these diseases are also leading causes of death. So in my lab, well, among this listed here, or we study heart disease, stroke, Alzheimer's disease, and Alzheimer's disease because they have a lot of shared etiology. Okay, so if you look at the prevalence, just take heart disease as an example. This is a US map. And you can see that it's quite, actually 1/3 of the population has heart disease. But it's more severe or more prevalent in certain places than the others. So that explain that, kind of raises the question why, and why do different people at different places might have different risks. So anybody wanted to say why that could be? Any guesses? Nobody want to guess? Oh,
Socioeconomic factors.
Socioeconomic state, yeah, yes. And also environment, diet, lifestyle, right? There are many things that could be involved. So indeed, that lead to, the complexity, right? Not everybody will carry the same kind of risk. There are different types of risks are involved in the complex diseases. Like we talk about genes a lot, definitely all these complex diseases have very heavy genetic component, and also the environment, right? Depending on the diet and the environment and socioeconomic status, there are a lot of things that could contribute to these diseases. And then, so for the heart disease here, I'm showing that the heart, actually the most common type of heart disease is not about the heart muscle, it's actually about the coronary artery, which are the arteries that supply the nutrient and oxygen to the heart. And the main cause is the blockage of these arteries. So where does the blockage come from? Actually come from the liver, adipose, and actually the brain can also regulate the functions of this arteries. So actually it's a lot more complex than we thought. The heart disease is just the heart. It's actually involving many other different places as well. So that brings to the second complexity that, not only there are many different causes and genes and environment, but also there are many different tissues and organs that could be involved in one disease. So here I'm showing you another disease, type 2 diabetes. So for that disease, brain, liver, muscle, intestine, even the fat tissue, they can all be involved in how our body regulates glucose control. So you have to realize that we can't just study one particular, organ or tissue. Most people think, okay, it's pancreas is the problem for diabetes. That is one of the main causes, but actually there are many other things involved as well. So that's make it a little bit more complex, right? So even within each of these tissues or organs, there are many different types of molecules. So we call them like omics. So I will introduce all these different layers of omics a little later. So there are different molecules. They actually interact in networks that are within tissue or also between tissues. So different molecules interact and regulate the tissue function, and also they can crosstalk between tissues. So the key here is actually there are main key regulators that could be more important than the other ones. The idea is how we can take all the data, all the information, and find this kind of networks and key regulators. So classic approaches to address these diseases, I think you already got a flavor. We can study one gene at a time, right? So you can you can do this kind of a study by knocking out a gene or adding the gene back. So for example, if we think about type 2 diabetes, I talked about all these tissues are important. And let's see, okay, what about I believe gene A is important, let me take it out or add it back, or gene B is important. We can do this, right? We can do screens. That might be easier to do it in a smaller organism like a Drosophila or worms, but actually in mice or bigger or model organism, it's harder to do. It takes forever to really understand that. So actually, they makes it. If we the idea is if we understand what each gene does in each tissue, then we can piece things together. But when we piece it together, it may not be exactly what we want it to be. So the challenge that we're facing with over the past few decades is that this breakthrough discovery for so many more genes for each of the diseases, the complex diseases we talk about. If you want to take a guess, what's the range of genes that you would guess that's involved in each of these diseases? Like in tens, in few, or hundreds, or thousands? What would you say? Anybody want to say something?
Tens.
Well,
Hundreds.
actually, yeah, hundreds. Yes, at least 100 that reach statistically significant cutoffs, right? If not thousands. As we increase the study population, we find thousands or even more. So that's what's making it challenging. Do we want to study, knock out every single gene and study every single one of them in every single individual tissue or cells? So that becomes a little bit inefficient. So so we kind of start thinking about a different way of looking at complex diseases. So this is a paper published in Cell in 2017, which bring out this concept of omnigenic disease model, which means that most of the genes are involved in these complex diseases. Because the genes are interacting in networks, and actually the network, the rules in the networks are not, all the same. Some are more important. We call them core genes or hub genes, and some are a little bit less important. They're in the peripheral, so we call them peripheral genes. Okay, so if you have to take a guess, which genes are more important, the core or the peripheral?
Oh, everyone's typing in the chat.
Yeah, everybody says core genes, great. Yes, exactly. So I think that kind of highlight our approach, right? So we don't really need to understand every single gene what it does. If we could really look at how they interact, we can prioritize certain core genes. Maybe that's more effective in looking at the disease. So okay, so I think that's my long background introduction on the multi-dimensional complexity of the human diseases, kind of pointing to some ways of we can do this kind of research. So this brings to the second bullet point here. What is system biology? How can it help us address all that multi-dimensional complexity we talked about? So this is a big data science. It's highly interdisciplinary, involving engineering, mathematical, statistical, computational principles to address biological questions. So you need to have some background in different areas to really be able to effectively do this. We focus on complex interactions, not the individual genes or component, but the complex interactions within the biological system. So we call it a holistic approach. So over the past decade or so, there has been very strong debate on the, which way to go. Do we, need to stick with the reductionist approach? Do we need to really move all the way to the holism approach? And the debate is still today. Actually, in fact, yesterday I just had a big argument with a big professor at UCSF because we're on the two different spectrums, and he thinks the approach that we're taking is making things more complicated, and that you will never solve anything, right? And my approach, my answer would be, well, we probably need to do both, right? So what I think is, if we do have a very strong belief, we wanted to find something, yes, the reductionism approach is very good. You can go really find your gold if you believe it's there. But the system biology approach is, I don't know. It's so complex. I just wanted to use the data to tell me what might be more important, right? I first look at the whole picture. I find something that's probably more important. Then I go back to the reductionism approach to really go find what I need to find. So to me, there shouldn't be debate. There should be just a harmonization of the different approaches, and we work together. One big picture, one a little bit smaller, kind of a more focused study. So put it in a bigger concept. So we will see that the system biology is just using networks of networks to delineate the complexity. So we really wanted to understand the molecular networks, all the genes, how they interact, and then there are many cell types. And how the cells communicate in tissues, and also how the tissues interact to really understand the complexity. So that's my introduction for the system biology part. So next I'll go through some of the studies on what we have learned about the complex diseases. So in my lab, we partition the diseases we study into two camps. One is the cardiometabolic side, so the heart disease, diabetes, obesity, etc., because they are highly connected cardiometabolic issues. And then the brain disorders from Alzheimer's disease, ALS, etc. So we also look for connections between different systems as well. So the approach we take, obviously, we need computational big data integration. 70% of the lab does that. And we do have a wet lab as well, because I do wanted to integrate the reductionist approach to really, once we narrow in onto some strong candidates, we can do experiments and really test them and see if they are real, right? And then the approaches we take, obviously, I have introduced earlier. So we take the different type of risk factors from big data and look for their effects on individual tissues, and go into the details on the different omics and look at how they interact and look for key regulators. All right, so here is the slide I wanted to go through these omics domains. I think for you guys, you must be very familiar with the DNA that goes to RNA, right? Goes to genes, and from genes that goes to the protein. So basically these three layers should be very clear to you. What I'm adding a little bit here is the epigenome, which is, the DNA sequence where we inherit them from our parents, but actually there could be epigenetic modifications like DNA methylation, histone modification. I'm not gonna go to too much detail, but there are different ways to modify the DNA, so you can turn different genes on and off, okay? Then the metabolome side is just small molecules like different fat molecules, amino acids, sugar molecules. There are different molecules that could fill in the molecular space. So there are intrinsic interactions and workflows across the different molecules. We can leverage that to understand the complex system. And there are also many, and I put the numbers here to just give you a hint on how big the data could be. So it's not like you can open a file with an Excel sheet, right? So it's going to be quite big data. So then from each layers of this, you can look for disease association. For example, I wanted to know on our DNA, we inherited from our parents, which pieces of the DNA is associated with the disease. So that would involve this so-called genome-wide association study, GWAS, that have become very powerful. And then if you look at the epigenome layer, you can do epigenome-wide association study called EVAS. And I'm sure you guys are all very smart. You can guess here. If I look at the transcriptome, what would that be? Need to TVAS. Yes, great. Yes, so TVAS, that's very good. And then you can go continue with that, and then protein PVAS, etc. Right? So you can really just look at what molecules are associated with the disease. Then the next task is, how do I piece all these things together to really understand the disease? Then we need to develop tools, right? So our lab, through the efforts of a few former postdocs and the PhD students, we have built this tool called Merge Omics. So Merge Omics, right? try to merge the different omics domains to really look for what kind of networks are perturbed by the different type of molecular signals. How do they interact in the network? And then find the key regulators. So we have our package you can download if you do coding. You can also use our web server, so you can upload your data and do your analysis using our tool as well. And that could help what Mergeomics does is it can help us narrowing onto disease networks and the key regulators. So we can also use this kind of network to ask, can I find a drug, an existing drug, for example, FDA approved drug, to see whether that drug, any drug can perturb this disease network, right? Then we can do drug repositioning. So to do that, we developed a second bioinformatics tool called Omics. So here we wanted to take all the FDA approved drugs and look for their molecular signature and in different species, in different tissues. So that way we can build a database to see, okay, a drug can affect these genes in the heart, in the human, or versus the other species. And then once we have this database, we can ask the question, if I have a disease network, so the genes highlighted in red here are the disease genes, and if I can find a drug molecule that hit the similar network, not necessarily the same set of genes, maybe that drug can be used for that disease as well, right? So that's kind of the concept we have here. Again, we have online tool that people can use. So over the years, we have been applying this kind of tools to study many different diseases. I'll just give a very quick example on fatty liver disease that we have recently did. So fatty liver disease is a very significant risk factor for heart disease, diabetes, etc. So it's basically a fat built in the liver, and then it causes inflammation, causes scarring of the liver. And the mechanism is not very clear, and there is no FDA approved drug to treat this condition, okay? So we still need to better understand it. So we collected a lot of omics data from collaborators, public domains, to try to model the network. So here we modeled 2 different tissues. So we included both adipose and liver because we believe those are the relevant tissues for fatty liver disease. And indeed, we find many different genes in many different processes seem to be very enriched for genetic risks of the liver disease. So in adipose, you have genes in insulin signaling, for example, in immune system, metabolism, etc. And there are also processes shared between tissues. And then we take these genes, we can really look at their interaction. For example, here is the liver network we modeled. Based on the genes that are involved in fatty liver disease, we could predict, therefore, key regulators seem to be really important in regulating all the rest of the genes. And now let's go to do experiment, right? If I take them, lower their expression level in the liver, do they really affect the liver buildup, the fat buildup in the liver? Indeed, by lowering the expression of each of these 4 genes, we could lower the liver fat. And we also validate that when we take these genes out, indeed, the neighboring genes in the network were all changing as well. So we could validate the network structure and also validate their function in fatty liver. And by the way, 2 of these are completely novel, which were later confirmed in additional human studies, okay? So that's kind of the power of this kind of approach. And another way of using the network, as I hinted, we could also look for drugs, existing drugs that target this network, right? So using the FDA approved drugs, using our Pharmomics tool, we predicted a few drugs which may really reverse the fatty liver. So we treated the animals, so he's the control mice that don't have increase in the liver fat, And then we treat, this is the disease animal that have increased the liver fat. We treat with one of these drugs, it actually significantly reduced the liver fat. The other one didn't work as well. It was it had a much lower rank in our prediction. So actually the rank in our prediction also seemed to matter. So that's another way of using the network to look for kind of potential therapeutics. So that was an example for one disease. We can also study, the relationship between highly connected disease like heart disease and diabetes. We know that they segregate together, but are there shared mechanisms? Again, we can collect a lot of data from the public domain or from collaborators and do our modeling. And again, we can find sophisticated networks, but there's a structure. So there are key regulators that organizing these disease genes in different tissues. So this part of the network is mostly coming from the fat tissue. This is mostly from the immune system. This is mostly from the hypothalamus of the brain. So this again raises new hypotheses so that we can go back to the lab to test how they are relevant. So that's another example. So the last part of the example I wanted to show is we are currently moving beyond just the tissues, because each tissue, there are many different cell types as well. For example, the brain has neurons, many different types of neurons, astrocytes, oligodendrocytes. So, but when we look at the tissue, we're grinding everything together, right? So now we have the technology to look at all the genes in every single cell. So we could do thousands, tens of thousands of cells at once to look at every single disease. We have been applying that to both cardiometabolic disease and brain disorder over the past 3 or 4 years. So for example, for a brain disorder, we studied traumatic brain injury. We analyzed close to 80,000 cells from 3 different tissues, two brain region and also the peripheral blood. So there are many different cell types. Each dot is a cell, okay? And each dot is summarizing thousands of genes in that cell, okay? And different tissues and different time points. I'm not going into detail. What I wanted to tell you is the power of the data that we could generate nowadays, and we can also use it to tease apart more detailed mechanisms in the disease. So for example, using the data, we could model how the cells communicate in the brain. This is the hypothalamus cell-cell communication map based on the data we generated. So this is in control animals. This are in animals with traumatic brain injury. You can look into more detailed interaction. For example, here between the astrocytes and neurons, these are the predicted interactions based on the data. And then in the disease situation, you can see the interactions are changing, and there are many different genes that are communicating between the different tissues. So kind of having more granular maps. So to summarize what we have learned here, this type of system biology can help us dissect the tissue and cell type disease mechanisms, and also can help us pinpoint the key regulators, and also potentially can help us find a new therapeutic agents to normalize disease networks. So the last 2 slides I'll focus on talking about the importance of bioinformatics and big data analytics in this area. Again, I bring up this particular slide to remind you the high dimensionality and the complexity of the data. So to do this kind of research, you do need highly interdisciplinary knowledge and skills. So you need to be able to design analytical algorithms and computing programs, and build databases and tools for yourself and for others. And on the system biology side, there are people who think they are the same thing, but I do not believe they are the same thing. I think system biology has a lot more of the biology component. So in addition to bioinformatics skills, and also I want to emphasize even experimental capacity, because just modeling the data, integrating the data is not the final step. You really need to show in experiment that they do work. So basically, I think this image from the Institute of System Biology summarizes, right? It's a field that we integrate biology, technology, and computation. We have biological questions to address, integrate new technologies, new data, and new software to generate new hypotheses and insights. Then we go back to the circle to eventually get to the full picture. With that, I want to thank all the people who are involved in the few projects that I showed here. So I actually acknowledged many of them on the slides when I introduced the project. Also acknowledge our funding from NIH. And my lab members here. So they definitely a very happy group of people, and we all, work, I guess, in a pretty pleasant environment, and really love their energy and all the contribution. Thank you, everybody, for your attention, and I'm ready for questions.
Thank you so much, Dr.
All
Yang.
right. Of course. All right. Let me see the chat. Questions? Do you take high schoolers? I did take one high school student so far. So yeah, the lab is a little bit saturated. So it's getting harder for me to give everybody attention. So my philosophy is that I need to give everybody who come to the lab enough attention. If they just come and I don't even have interactions with them, I feel like I'm failing them. So, if everything matches, and if you are very enthusiastic, we do have kind of senior student trainees who can help give you more guidance. I'm happy. And what do you mean by experimental capacity in systems biology? Oh, what I mean is you need to be able to do experiments, to actually design experiment to test what you found, and know how to do the experiments as well. So that's what I mean. Okay, what else? Has any of your work led to subsequent reduction follow-up work? Yes, so I showed in the fatty liver example where we found 4 predicted genes, and we went back to the reductionist approach. We take them out, right? Take the gene out in the liver and look for downstream mechanisms. And what we found was that 2 of those affect mitochondrial function. So we do go down to, dig into the more detailed mechanism for individual genes. Yeah. With COVID-19, is your lab open in fall? We're open all the time because it's mostly online, right? As I said, 70% of the lab is computational. So all you need is a computer. You can access to our big computing cluster, log in, and we have been working remotely from mid-March. So yes, the wet lab side has reduced the capacity for sure, but the dry lab is fully open. What are the biological or biggest challenge you face in leading such a diverse team? Actually, I didn't feel a lot of challenges. I find that it's a lot of fun to work with people with very different background, because they can, everyone brings a very unique perspective. They can teach me a lot new things. I feel excited about it, and I don't feel like intimidated because, science is constant learning, right? You learn, you improve, and just because they are your trainees doesn't mean they know less than you. Actually, many of them bring very new techniques here. So I don't feel very challenging. I do feel sometimes that I can't guide certain directions because I lack that knowledge. So I do try to build connections with more collaborators and things like that to help bring in more expertise to help guide the trainee. Yeah. Can you expand on epigenetics? Epigenetics, it's so I guess it's more of I would see a modification or reorganization of the genome. So one most common type, I don't know if you guys know, is the DNA methylation. So on the cytosine, on the C, letter C's of your ATCG sequence, you can add a little methyl group, and that will change the function. That will change whether the gene is gonna be turned on or off. That's only one type. The epigenomic field is so broad that, you know, I can give you a full lecture on that. All right, I see many more questions. Proficient in Java, Python, MATLAB, and some other. Oh, that's good. That's great. That sounds like you have a full package. If you know R, that would be even better. All right. And the helpful resources for students who are interested in this area of research. So there are a lot of online classes nowadays on bioinformatics, essential tools like Python, MATLAB, R, you know, Linux, you know, SQL databases, etc. So yeah, anyway, I think I'm taking too much time already. I don't want to go overboard. I hope I answered most of the questions. Thank you so much.
Sorry, give me a sec. Okay. Yeah, thank you so much, Dr. Yang, for joining us.
Of course.
It was really awesome to get to listen to your talk about systems biology.
All right, great. Thank you. Thank you, everybody.
Yeah. Right now, we'll just have a 5-minute break until the biology competition. So if you're not competing, feel free to leave for now. And we'll resume talks at 12 p.m.
Bye, everyone.
Okay, thank you.
Bye.
Oh, we're staying on this session. Okay, yeah.